now.
I would request all the teachers to switch on their video. The departmental teachers, please switch on your video.
Good evening, one and all. Welcome to the 17th chapter of Modern Trends in Microbiology, organized by the Department of Microbiology of St. Xavier's College, Autonomous, Kolkata. MTIM is an annual seminar of our department which aims in expanding the knowledge of students about recent advances in the field of microbiology. Due to the current COVID scenario, we have chosen to conduct this seminar as a webinar in an online platform. We would now like to begin with the inaugural ceremony with Holy Scripture reading and an opening song. The Holy Bible will be read by Romy Rishid George of BSc 3rd semester. The Holy Quran will be read by Sheikh Shahaban Badshah of BSc 3rd semester. Srimad Bhagavad Gita will be read by Orgopriya Bhattacharya of BSc 5th semester, followed by the opening song by Tanishtha Bishash of MSc 3rd semester of Department of Microbiology.
A very good afternoon to all of you. The Bible passage I'm going to read today has been taken from Psalms chapter 40, verses 1 to 5. I waited patiently for the Lord. He turned to me and heard my cry. He lifted me out of the slimy pit, out of the mud and mire. He set my feet on a rock and gave me a firm place to stand. He put a new song in my mouth, a hymn of praise to our God. Many will see and fear the Lord and put their trust in him. Blessed is the one who trusts in the Lord, who does not look to the proud, to those who turn aside to false gods. Many, Lord my God, are the wonders you have done, the things you have planned for us. None can compare with you. Were I to speak and tell of your deeds, they would be too many to declare. Thank you.
Good afternoon, everyone. So I'll be reading out Surah Al-Baqarah, verse 155 from the Quran. Bismillahir Rahmanir Rahim. وَلَنَبْلُ أَنَكُمْ بِشَيْءٍ مِّنَ الْخَوْفِ وَالْجُوعِ وَنَقَصٍ مِّنَ الْأَمْوَالِ وَأَنفُسِ وَالثَّمَرَاتِ وَبَشِّرِ الصَّابِرِينَ الَّذِينَ إِذَا أَصَابَتْهُمْ مُصِيبَةٌ قَالُوا إِنَّا لِلَّهِ وَإِنَّا إِلَيْهِ رَاجِعُونَ. This verse states, "We will surely test you with something of fear and hunger and a loss of wealth and lives, but give good tidings to those who are patient and have sabr. Those who say when afflicted with a calamity that we belong to God and to whom we shall return are the ones who will have blessings and mercy from their Lord." Thank you.
It was the war of Kurukshetra. The thought Pandav Arjun, with an agitated mind, was unable to realize what responsibilities he was indebted with. He was in a dire need of consolation, comfort, and was craving for light, being engulfed in the murky tunnel of worldly pursuits. Lord Krishna chose to impart supreme knowledge to his beloved Partha. In the second chapter of Bhagavad Gita, namely Sankhya Yoga, or the Yoga of Analysis, he told, Ya enang vekti hantarang yashchainam manyate hatam uvo tau nabiyanita nayam hanti nahannati, which implies he who holds that soul can be slain, or he who considers that the soul can destroy, both of them are ignorant and plain, while soul neither slays nor it is slain. वसांसि जीर्णानि यथा विहाय नवानि गृह्णाति नरोपराणि तथा शरीराणि विहाय जीर्णान्यानि संयाति नवानि देहि. As a person sheds worn out garment and wears new one, likewise at the time of death, the soul casts off its worn out body and enters a new one. So neither we have the power to destroy the soul, nor it is slain. We always should be concentrating on our own duties. As the Lord says, Karmanyavadhikaraste ma phaleshu kadachana. We have a right to perform our prescribed duties, but not entitled to the fruits of our actions. Thank you.
Sangachadam Sangbadatvam
शम्भु मनांसि जानता संगच्छदं संबदाध्वं शम्भु मनांसि जानता समानो मंत्र समिति समानी समानो मंत्र समिति समानी समानं मनसः चित्त में संगच्छदं संबदाध्वं शम्भु मनांसि जानता. Thank you.
Thank you so much, all of you, for such a soulful beginning to our webinar. I now request Professor Dr. Arup Kumar Mitro to request our Father Principal, Reverend Dr. Dominic Savio SJ, to give his welcome speech and release the 7th volume of our departmental magazine, The DNA, that has been beautifully collaborated by Shobhik Banerjee, Vaibhavi Bhattacharya, Spondon Mojumdar, and Piyash Mukherjee.
Thank you, Angshu, for the introduction. This year, the 17th chapter of Modern Trends in Microbiology is a little bit different from the other years because we are meeting in a virtual platform and we have the speakers all from abroad. And definitely this year also we cannot have the poster presentation, but instead we have kept two types of presentation, one paper presentation and one short communication. So the short communications will be like poster presentation. And I could see that around 20 participants from other institutions have been shortlisted. And there will be around 65 different presentations in the 3-day tenure. And there will be 4 speeches by our respected guest speakers in different aspects of microbiology. And of course, there will be some lectures associated with the topic of COVID, the present day rat that we are facing. So with these words, I would request our beloved, respected principal, Reverend Father Dr. Dominic Savio, to give the welcome address and also release our 7th volume of DNA. We call it eDNA this time because it is only in e-version that this magazine will be circulated. Over to you, Father. Thank you.
Thanks, Professor Arup Kumar Mitro. Professor Sudeshna Saim Chowdhury, the head of the Department of Postgraduate and Research Department of Microbiology. Professor Dr. Arup Kumar Mitra, the convener of 17th chapter of Modern Trends in Microbiology. Dr. Rajyashri Sen, the guest speaker. All the respected faculty members, guests, and our dear students. It gives me great pleasure and pride to present before you the 17th chapter of Modern Trends in Microbiology 2020. The Microbiology Department of St. Xavier's College has always been in the forefront of cutting-edge research, and this is due to the endeavor of both the students as well as the faculty of the Department of Microbiology. This event has now grown as much awaited annual event among the students of this college, as well as from others throughout India. The seminar has a tradition of inculcating in the students a sense of appreciation for research and the enlightenment of them with a know-how about different new progresses, about the current advancements in the field of ever-developing science. This year, there are many troubles we are facing due to the ongoing pandemic, as well as the other day-to-day problems often encountered in the new normal conditions. But this did not stop the students and the faculty members of the microbiology department of the college to organize this event in digital platform. I hope we encounter no problem during the conduct of this event and it runs smoothly. I hope students from all universities and colleges, including students of our college, will find this seminar informative with a scope to learn more and hopefully in this current scenario of pandemic will provide a proper platform to know more about the modern trends in research without leaving the comfort of home. I hope this will disseminate enough information to every student out there and will be a successful event together. With these few words, I congratulate the Department of Microbiology, the head of the department, Professor Arup Mitra, and all the faculty members and the students, our dear students, for organizing this 3-day international webinar. And I wish you all the best. May God bless you.
Thank you, Father.
Yeah.
Thank you.
Thank you, Father. Thank you, sir. The e-version of the DNA will be circulated shortly in your respective WhatsApp groups. I would now like to invite Professor Dr. Sudeshna Sham Chaudhuri, the HOD of our department, to introduce the audience to modern trends in microbiology.
Just a moment. Father will release the eDNA before it is circulated. Please, Father, would request you to release the eDNA.
I don't know whether people are able to see this one.
Yes, Father. Thank you.
Able to see? I don't know.
Thank you, Father.
Okay. Wish you all the best.
Thank you.
Thank you. Thank you very much, Father.
Yeah.
Thank you.
Yes.
And I would like to welcome our coordinator IQAC, Dr. Parthopratim Ghosh, and our Dean of Science, Dr. Tapati Dotto, to the platform. Thank you very much for joining.
Thank you, sir. Thank you, sir. Thank you.
So over to Sudeshna ma'am.
Thank you, Dr. Mitro. Good evening, everyone, and a very warm welcome to all of you to Modern Trends in Microbiology, Chapter 17, 2020, organized by the Department of Microbiology, St. Xavier's College, Kolkata. I would like to send my sincere greetings to Reverend Father Dr. Dominic Savio S.J., Principal, St. Xavier's College, Autonomous, Kolkata, Professor Bertram Da Silva, Vice Principal, Arts and Science Department, Dr. Tapati Dotto, Dean of Science, Dr. Arghu Banerjee, Dean of Arts, Dr. Ayan Chandra, Member Secretary, Academic Council, Dr. Parthopratim Ghosh, the IQAC Coordinator, all of our esteemed honorable guests, the associate and the assistant professors of our department, my dear research scholars, my dear students, and all internal and external participants. I'm glad to take this opportunity to welcome you all to the annual event of the Department of Microbiology. Department of Microbiology is the department focusing on practical application of theoretical knowledge. Maintaining a good teacher-student bonding, it tries producing scientific papers, keeping in mind the broader aspect of life. The professors and scholars being the support system of practical knowledges, the approach to science is transdisciplinary, giving equal emphasis to biostatistics, biophysics, biochemistry, and bioinformatics, including classical and applied aspects of microbiology. Out of many international initiatives like Frontiers in Biological Sciences 2020, a two-day webinar conference on scientific and psychological aspects of COVID-19, today we have gathered for the three-day international webinar, Modern Trends in Microbiology, Chapter 17, a level higher, even under this current threat of pandemic. I would like to wish the best to the students who worked day in and day out for putting up this three-day endeavor through science and culture. I would also like to congratulate them in advance for all the success. This year, we are happy to host our ex-students as our guest speakers who are involved in cutting-edge research serving the bigger society in their respective esteemed laboratory abroad. We are indeed very lucky and delightful to have as our speakers Dr. Shamantrita Bhattacharya, postdoctoral research fellow of UTSW Medical Center, USA, Dr. Rajyasree Sen, postdoctoral research scientist of Excel Laboratory, Zuckerman Institute, Columbia University, Dr. Bharat Vaidyanathan, senior scientist, exploratory immunology, EMD, Serrano R&D Institute, USA, Dr. Nandini Haldar, Senior Registrar, Department of Obstetrics and Gynecology, Huel DDA University, Wales, UK. We are hopeful that this webinar will turn out to be an effective and significant one in our current scenario. Finally, on behalf of entire teaching fraternity of the department, I would request Father Principal and our honorable esteemed guests present to bless all the organizers and students for the successful journey through modern trends in microbiology, chapter 17, 2020. So once again, I want to convey my regards to Father Principal, Vice Principal, Dean of Science, Dean of Arts, Member Secretary Academic Council, and IQAC Coordinator. I want to welcome you all. Thank you.
Thank you, madam. Thank you very much.
Thank you, ma'am, for the amazing introduction. I now would like to request Professor Devjani Datta to give a vote of thanks and end the inaugural ceremony.
Okay. Thank you, Anshu. Good afternoon to all present here. It is my proud privilege today to confer the vote of thanks for the inaugural session of the Modern Trends in Microbiology, Chapter 17. I, on behalf of the entire microbiology family, extend my heartfelt thanks to our beloved principal, Reverend Father Dr. Dominic Savio, for his encouragement and support all throughout. I take this opportunity to thank our Vice Principal, Sir, Professor Bertram Da Silva, the Dean of Science, Dr. Tapati Doctor, and Dean of Arts, Dr. Orgho Banerjee, for constantly being with our department in all our endeavor. Thanks are also extended to the IQAC coordinator, Dr. Parthraputim Ghosh, and Member Secretary of the Academic Council, Dr. Oyun Chandru, for their constant support and encouragement. A big thank you to Professor Romit Beed and Professor Jayati Ghosh Toshtidar of the Computer Department, St. Xavier's College, for coordinating with us in providing us the Zoom platform for conducting this webinar. I cannot thank enough our head of the department, Dr. Shudeshna Sham Chaudhary, and the convener, MTIM, Dr. Arup Kumar Mitro, without whose constant and tireless efforts, this webinar could not have been realized. Last but not the least, I would extend my thanks to the entire Department of Microbiology, the MTIM organizing committee members, and the entire audience of MTIM who will be present on this auspicious occasion during the three-day webinar and make it a success. Thank you, one and all present over here.
Thank you, ma'am. We shall now proceed to the first technical session of the day. I would request Professor Dr. Kasturi Sarkar to introduce our first guest speaker, Dr. Rajoshri Sen. Over to you, ma'am. Ma'am, you're muted. Kindly unmute your mic. Thank you.
Am I audible now?
Yes, ma'am, you're audible.
Hello, everyone. It is almost 5 p.m. in India, so I am saying good afternoon to all of you attending this webinar. I have got the pleasant duty of introducing our today's guest speaker, Dr. Rajashree Sen. It is always a proud moment for a teacher to tell about his students' achievements to others. And I am immensely happy to welcome Dr. Sen and to introduce her to you. Welcome, Rajashree. Welcome to your alma mater. Rajashree completed her graduation from Department of Microbiology, St. Xavier's College, Kolkata in 2010. And she did her post-graduation from University of Hyderabad. Then she did her doctoral studies in Research Institute of Molecular Pathology, Austria, and HHMI Genelia Research Campus, USA, from 2012 to 2017. And her doctoral research topic was neuronal control of backward walking in Drosophila melanogaster. She continued her postdoctoral studies in HHMI for 1 more year, and currently she is associated to Zuckerman Institute of Columbia University and working on neuronal representations of social memory in mice. Rajashree has received many fellowships, like the fellowships of Indian Academy of Sciences and Department of Biotechnology during her graduation and post-graduation. She got the Marie Curie Research Training Network LIAC Fellowship during her doctoral work. She has published many research articles in peer-reviewed journals having high impact factors. And from my personal experience, I can say that I found Rajashree in her graduation days as a fun-loving, jolly individual who loves to do many extracurricular activities. At the same time, she is very serious about her studies and career. I believe her journey will inspire all of you students present here attending today's seminar. So today she will be discussing on nervous system control behavior. So Rajashree, over to you.
Thank you. So I'll share my screen. Just a second. I think. So first of all, I want to thank all the organizers. First, congratulate all the organizers for putting this together. And thank you for inviting me. It's really an honor to be here presenting in the Modern Trends in Microbiology seminar. And it makes me very emotional because I think around a decade ago, I was on the other side of this conference, and it was events like these that made me very interested in research. And I must say that it has been a very adventurous journey. Whatever I wanted to ask had taken me to different countries, and I've worked with a lot of people from all over the world. And I think one of the most gratifying things of doing research is kind of being the first person to see something. And no matter how small it is, it's kind of like a very gratifying moment. So I hope in my talk today, I can give you a flavor of what that process is to kind of like do research and be a part of this, ongoing endeavor of scientific contribution. So today Can you see my screen?
Yes, doctor, we can see.
Okay, so today I'll tell you a story about some neurons that switch fruit flies into reverse gear. This is my graduate work, and I did this work in the lab of Barry Dixon at Genelia Research Campus. Animals usually walk forwards, but sometimes they also walk backwards. So here's a reason why an animal might want to back up. So just like this cat, which is really scared, it is this backing up behavior is kind of an avoidance behavior. And it is as instinctive in a cat as it is in a human. So think of the last time you were about to cross a road and the pedestrian sign switched from green to red. You perhaps backed up into safety. So how do our brains control these avoidance maneuvers in response to visual threats? This is a question which is really hard to address in a human brain because the human brain has around 100 billion neurons. And with the current tools that we have, we can at most kind of say what brain region controls what behavior, but we do not have the tools to monitor or manipulate the human brain at like cellular resolution, which means that the way a brain works is not in, you know, it is in kind of in single neurons control behaviors, and we do not have the ability to manipulate or monitor single neurons in a human brain. So how do we study this problem? So I want to show you a video of a praying mantis. Here's a praying mantis in presence of a fruit fly. And when the praying mantis turns its head in the direction of the fruit fly, the fly backs up and retreats. So this avoidance behavior in response to a visual threat is as instinctive in a human as it is in a fruit fly. Now the question is, how does a fruit fly brain process this visual threat and convert this visual threat into an appropriate motor action, which is retreat? Like, how does it see a threat, feel the fear of the threat, and generate this very specific output, which is retreat by walking backwards? Now, this is a more tractable question because the fruit fly brain is monumentally simpler than a human brain. A fruit fly brain has around 250,000 neurons. And you can, with current tools, we can literally, we can change the DNA of a fly so that we can turn on or turn off as few as one neuron in the entire brain of the fly to figure out how that neuron controls behavior. And this kind of experimental control is simply not available in more complex organisms. And even 10 years ago, this was not possible in a fruit fly. So just like, you know, technologies like in DNA, advances in DNA technology or advances in forensic technology allow us to solve crimes that we could not solve 10 years ago. There has been excellent breakthroughs in neuroscience, technological breakthroughs in neuroscience, which allows us to address questions that we could not have addressed around 10 years ago. And one of those techniques, which was really indispensable for my research, was called optogenetics, which is a way to use light to turn on individual neurons in the nervous system of the fly. So literally, I can shine light on a fly, and it turns on as few as one or two neurons in the entire nervous system of the fly, generating a behavior. So that I can correlate that particular neuron is causing a specific behavior. So the way it works is you change you do this transgenic, you generate this transgenic fly. So you change the DNA of the fly so that small subsets of neurons in the nervous system of the fly contains a light sensitive neuronal activator or inhibitor, so that when I shine light on these flies, only those neurons turn on. And this results in a change in behavior, so that I can correlate that those particular neurons result in the specific behavioral phenotype. So before I joined the lab, there was a graduate student who was doing these experiments in flies. So it's, you can think of it as almost a genetic mutation experiment, but for neurons, just like when you mutate genes to figure out what gene controls what phenotype. He was switching off and switching on neurons to figure out how those neurons control behavioral phenotypes. And so he was doing this screen, this neuronal activation screen, where he was activating small subsets of neurons in the nervous system of the fly, looking for neurons which, when activated, result in interesting behavior. And he found this fly. It's a fly that just walks backwards. And at the time, it was surprising, because when do flies walk and when do flies walk backwards? So this was a very kind of interesting avenue. But it was also really cute, because it was the fly's version of Michael Jackson's moonwalk. So we affectionately call this fly the moonwalker fly. And the neurons that control this behavior as the moonwalker neurons. So what I'm showing you here on the left is a fly. Inside the fly is a cartoon of the nervous system in yellow. And here's just an expanded view of the nervous system. So on top is the brain of the fly. And on the bottom is the nerve cord, which is the fly's equivalent of a spinal cord. And in green are stained neurons, the moonwalker neurons, on the brain and the nerve cord of the fly. So what you can immediately notice is that neurons don't look like regular cells, which are just round balls. They have these interesting processes which indicate regions where they receive inputs from and regions where they send outputs to. So in the case of the moonwalker neurons, the neurons receive inputs in the brain and send outputs in the nerve cord, that is the spinal cord analog of the fly, which controls the movements of the legs of the fly. Now, there is one thing which is, if you remember the video I showed you about a fly retreating from a praying mantis, it seems like there is a visual cue that triggers the fruit fly to retreat. So the mantis looks at the fruit fly, and the fruit fly looks into the eyes of the mantis. And then that is the critical moment where the fly retreats. But the moonwalker neurons are a collection of centrally located neurons. So they're located quite centrally in the nervous system, which means that they're not located at a region in the nervous system that handles vision. So there has to be some more pieces to this puzzle. So some neurons have to convey the information of visual threats to these neurons. So what are these neurons? So here is another piece of information that we knew. There were these neurons, visual neurons called the LC16 neurons that are located in the region of the brain which handles vision. And when these neurons are turned on, the flies also walk backwards. So what you're seeing here is a video of a fly that's walking forward. That is the green trajectory. And then we shine light on the fly. And when we shine light, these flies are genetically modified. So they contain a light sensitive neuronal activator in these LC16 neurons. So whenever we shine light, these neurons are active and the fly goes backwards because these neurons control backward walking. The other piece of information that you want to know about that is important about the LC16 neurons is that these neurons kind of respond to scary shadows. And a fly, when it encounters a moving like shadow, it's almost like the loom of a predator. And so this triggers a fear response in fruit flies, leading to backward walking, retreat. So far, this is the information I've told you. The moonwalker neurons are a collection of neurons in the central part of the nervous system. They're actually two pairs of neurons. They bring information from the brain of the fly to the nerve cord, which is the spinal cord analog in the fly. And when these neurons are activated, the fly goes backwards. I told you about LC16 neurons. These are visual brain neurons. They're located in the part of the brain which handles vision. And when these neurons are activated artificially, then the fly also walks backwards. So the obvious question that I wanted to test during my PhD was whether the LC16 neurons act via the moonwalker neurons to initiate backward locomotion, whether they lie in this pathway where the moonwalker neurons receive the information of visual threats from the LC16 neurons. Now, if this is true, so there are two possibilities, really. The first possibility is this hypothesis is true, that the LC16 neurons somehow talk to the moonwalker neurons, conveying the information of visual threats to the moonwalker neurons, triggering backward walking. Or the other possibility is that the LC16 neurons act via a different set of neurons to trigger backward walking, and the moonwalker neurons are kind of in a parallel pathway, triggering backward walking maybe through some other cues, like non-visual cues. So we tested these two possibilities. So if possibility one is true, then the simplest way this could be true is if the LC16 neurons and the moonwalker neurons are directly connected. Now, what you're seeing here is a three-dimensional rendering of a fruit fly brain. In blue are the moonwalker neurons, and in green are the LC16 or the visual neurons. So both these neurons trigger backward walking if they're artificially activated, in the fly if they're artificially activated. But what you can clearly see is these neurons are not directly connected. So they do not directly talk to each other. So, however, it could still be that these two neurons are somehow acting together to trigger backward walking via some other intervening neurons. So in an indirect way. So if that is true, then if we activate the LC16 neurons, this would trigger backward walking in flies. If we activate the LC16 neurons and then shut off or turn off the moonwalker neurons, then the flies should not be able to walk backwards anymore. However, if this is true, then if we activate the LC16 neurons and inactivate the moonwalker neurons, the flies should still be able to walk backwards because the moonwalker neurons kind of lie in a separate parallel pathway. So we tested this hypothesis. So on the left, you're seeing a fly where the LC16 neurons are just turned on, which is what I just previously showed. So the fly walks forwards, we shine light on the fly. This activates the LC16 neurons in the brain, and then the fly walks backwards. And here, we have a fly where the LC16 neurons are turned on and the moonwalker neurons are turned off. This is what we call a neuronal epistasis experiment. And you see that the fly is kind of stuck in this weird like rut. So it cannot walk backwards anymore. It kind of spins around and it's just the ability to walk backwards is gone. So it seems that the moonwalker neurons are absolutely essential for the fly to execute backward walking. And the LC16 neurons are important for, the LC16 neurons kind of act via the moonwalker neurons to trigger backward walking. So the moonwalker neurons are essential for the LC16 neurons to trigger backward walking. So it should rely downstream of the LC16 neurons. There's one more thing you need to know about these neurons. So you might have seen this interesting difference. When we turn on the moonwalker neurons alone, then the fly walks straight backwards. Whereas when we turn on the LC16 neurons, which means that the moonwalker neurons are active because it's upstream of the moonwalker neurons. Then the fly kind of walks backwards and then turns. Now this turn is, I think, really important for the fly because it helps the fly to reorient to a different direction, away from the predator, and otherwise it'll just move back to the jaws of the predator. But then how does the fly do this? So the first clue is, so the turn could be explained by some combined activity between LC16 and the moonwalker neurons. The second clue probably lies from the anatomy or from how the moonwalker neurons look like. So there are 4 moonwalker neurons, 2 neurons on each side of the nervous system of the fly. And what we have so far done is we have activated all 4 neurons on both sides of the body. What we have not done is activated 2 neurons on one side of the thorax or one side of the nervous system. And it could be that activation of the moonwalker neurons on one side of the nervous system triggers backward walking, longer backward walking strides on that side, leading to a turn. So if this is true, so we kind of tested the hypothesis. What we saw was that on the left you see a fly where all 4 moonwalker neurons are turned on. And in this case, the fly goes straight backwards. So the blue line is before the light is on. The red square and the red trajectory signifies when we shine light on the fly. And this is when the neurons are turned on. So when the 4 moonwalker neurons are turned on, the fly goes straight backwards. Whereas when 2 moonwalker neurons are turned on one side of the nervous system, the fly turns. So it seems that the turning can also be generated by or the turning is generated by kind of a patterned activity of the moonwalker neurons. So by activating the moonwalker neurons in a specific pattern, we can generate the flies actually turn. So I want to summarize what I showed you in this part. So this is what we think the model is. When a fly sees a visual threat, it usually sees it from one side or the other. More often the threat comes kind of not symmetrically, but from like either the left or the right. And when this happens, if a threat comes from the left side, it kind of activates the left LC16 neurons more than the right LC16 neurons, which in turn activates the left moonwalker neurons more than the right moonwalker neurons. And this results in a longer backward walking strides on one side of the body, leading to a backward turn, so that the fly reorients kind of to a different direction and moves away, kind of walks off or runs off at a different direction from where the predator is, from where the threat is. Now, I've talked to you about how visual threats trigger backward walking, but is visual threats the only cue that triggers backward walking in flies, or are there more additional aversive cues that could also initiate backward walking? If you think about us, people, think of the last time you didn't really know where you were going and you bumped into something. Probably your first reaction was to back up. A very reflexive, instantaneous reaction. Now, it turns out that flies also back up when they meet a dead end. So in this experiment, we put these flies in this really narrow grooves, so they kind of only walk forward. And in the end, when they hit the end of these narrow little chambers, they are forced to back up, and then they walk backwards. Now, the question is, how interconnected is the circuit that controls this touch evoked backward walking? How much of this is shared with the circuit that triggers visually evoked backward walking? So it turns out that if we turn off the moonwalker neurons in this assay, then the fly kind of gets permanently stuck in these narrow chambers. It tries to maneuver out of it, but it just cannot. So it kind of like stays stuck forever. So it seems that there is some interconnectivity between the circuit that triggers visually triggered backward walking versus touch triggered backward walking. And at least the moonwalker neurons are shared in between these circuits. The moonwalker neurons are absolutely critical for the fly to initiate backward walking. In the absence of it, it cannot. Now, in the previous section, I showed you about some visual neurons that convey information of visual threats from the environment to the moonwalker neurons. In this case, it seems that there is a touch seems to be the modality, which is the information that is being relayed to the moonwalker neurons to trigger this kind of backward walking. So we have found another set of neurons, which kind of relays the information of touch from the body walls and processes that and sends this information to the moonwalker neurons. And this is called, I identify these neurons, they are called the two lumps neurons. So these neurons have inputs in the ventral nerve cord, which is the fly's equivalent of the spinal cord, and which senses touch of the body walls. So whenever the fly hits a wall with its body, whenever it's kind of like and collides with a wall, it kind of touches with its forelegs and also with its body. And this information of touch is relayed to these two lumps neurons. And then these two lumps neurons, so here what you're seeing is the nervous system of a fly. This is the brain of the fly. This is the spinal cord analog or the nerve cord of the fly. In magenta are the two lumps neurons, and in green are the moonwalker neurons. And it seems that the two lumps neurons are very close to the moonwalker neurons. So they carry the information of touch to the moonwalker neurons. Now, what happens when you turn on the two lumps neurons? The fly walks backwards. So here again, the red trajectories are when the light is on, which switches on the neurons in the fly. And now the question is, do these neurons depend on the moonwalker neurons to trigger backward walking? So we did this experiment where we activated the two lumps neurons and we inactivated the moonwalker neurons. And it seems that the flies are again like unable to walk backwards. They kind of spin and try to walk backwards, but just cannot do that. So it seems that moonwalker neurons are critical for triggering backward walking, and they receive the information of touch from two lumps neurons and the information of visual threats from a different neuron, the LC16 neurons. So with this, I'm going to quickly summarize my work. So so the first thing that you have to know is that the key element that is extremely crucial for a fly to walk backwards is the moonwalker neurons. If we activate these neurons, the fly triggers a forward walking fly, or a fly that is stalled starts walking backwards. If we switch off these neurons, the fly is no longer able to walk backwards, whether it's in presence of a visual threat or whether it is in a narrow groove where it can no longer maneuver out of a dead end. So the interesting thing that you have to know is this is as few as 2 pairs of neurons. So there are 4 neurons. These are 4 neurons which we can specifically manipulate within a nervous system that has 250,000 neurons. And this experiment is impossible to do in a human. Even if we could activate as few as 2 neurons in the human brain, that wouldn't result in a behavior. So this is a very good starting point to understand how neurons control behavior because of the tractability and the simplicity of this kind of a nervous system. So using this as a starting point, then I found that visual threats are relayed by a group of neurons called the LC16 neurons, when the fly encounters a visual threat, this leads to asymmetric activation of the LC16 neurons. So if the threat is coming from the left side, the left LC16 neurons are more active than the right neurons. And this leads to a backward turning because it results in a longer backward strides on one side of the body. And then I also found another set of neurons, the two lumps neurons, that triggers, that kind of processes touch, that receives information of touch from the body wall. As the animal navigates through a rugged terrain, it can come across obstacles, and this results in, a kind of a signal in touch sensors in the body, which turns on the two lumps neurons and activates the moonwalker neurons, leading to backward turning. One thing is that these are three pieces of the puzzle, but obviously the circuit is very complicated and interconnected and complex. And this is something that we're still working on to kind of understand, to characterize the circuit in more detail. And one thing to appreciate about grad school is that we do not get to answer completely. We don't get to answer, we don't get a complete answer to our questions. We get small pieces of the puzzles. And then it's an effort of many labs with many people that gives us a comprehensive understanding of how circuits work. And so after I left the lab, a postdoc in the lab has followed up on my work and has actually found the circuit in the nerve cord that receives information from the moonwalker neurons and executes backward locomotion by activating muscles and motor neurons. So why is this circuit so important? So one of the things that one needs to appreciate about insects is that how flexible they move through rugged terrains. And many of and these kind of how the nervous system of a fruit fly solves the problems of navigating through rugged terrain has been a really inspiration for smart robotics. So one of the things that self-driving cars need to do is to avoid avoid accidents, is to actually process visual cues and tactile cues to avoid to kind of navigate through terrain. And we believe that inspiration from flies on how a fly solves these problems using their nervous systems would actually help make better technologies for these self-driving cars. So that is more of the application aspect of how this research. But I think in the end, the thing about fundamental research is to use the system, use a simple system as a fruit fly to kind of believe that at some point we hope that this would give us some insights on how we run away from a visual, like from a car coming towards us, or how we reflexively back up after hitting an obstacle in our way. And this is just a model system to understand how nervous systems work in general, because what nervous systems really do is to perceive the world around us, and then to quickly make a reaction, to quickly react to that, and kind of generate a motor output that is appropriate and goal directed. So, this is what we do every day. Our nervous systems take in information from the world and very quickly makes a reaction that saves our lives. So with this, I'd like to thank my PhD lab, the Dixon lab, which was in Vienna for a year and then moved to the US after a year. So it was a very international experience working with so many people from different countries. So I would like to especially thank Kai-Yu Wong, who did some of the experiments in the two lumps part of the story, the touch part of the story. And I would like to thank Bushke's lab in Cologne, where I did some experiments. And then I would like to thank my thesis committee and Barry Dixon, who was my mentor during my PhD. And with this, I'll leave you with a backward walking fly, which is headless. Thank you. and I'll be happy to take questions.
I now would like to request Professor Dr. Kasturi Sarkar to give the questions that has been sent via the chat box to Dr. Rajoshri Sen. Thank you.
First of all, I want to thank and congratulate Rajoshri for the wonderful, nice talk. Really simply beautiful. The way you presented your work with simple but very intelligent small experiments, it's really it's really very nice. And I believe that all the students attending this webinar have understood what Dr. Shane tried to tell you, right? So the questions. Why should I look for the questions? Okay, I have got the questions. So the first question is, can the moonwalker neurons of the flies be controlled by us?
Yeah. So that is what we did. That is what I did in my experiment. So what we do, so this is a genetic, with some clever genetic tricks, you can actually only express the light sensitive neuronal activator in the moonwalker neurons. So none of the other neurons in the nervous system have this neuronal activator, only that neuron has it. And then when we turn light, when we shine light on the fly, then those neurons turn on and the fly walks backwards. And the way this is done is by, we kind of have these large libraries of lines where kind of enhancer libraries, which express GAL4 in small subsets of neurons in the fly. And when these GAL4s kind of, when you cross these flies to other flies that contain, that kind of are transgenic and have a UAS neuronal activator, then only those neurons kind of express these neuronal activators. So yeah, so we're actually controlling, it's almost like remote control of neurons. We can switch on a light, we can shine light on the fly and know exactly which neuron gets turned on and exactly what behavior the fly executes. So yeah, it's really that, it's really that level of control that we have now.
The second question, you have already answered it. How do we activate the neurons artificially? If such activation is possible, then would we be able to activate neurons that are damaged and could be able to cure some neurological disease? How to turn on and off the neurons?
Yeah, that's a very good question. So you can do this in fruit flies, but it's hard to do it in how. So right now I work with mice. And then to some extent, we have some of these tools in mice where we can kind of inhibit or activate small subsets of neurons in the mouse brain. Like you can go into the mouse brain and stimulate a certain area and the mouse can start walking forwards or backwards. Or you can make it do, like it starts eating without stopping, so it becomes really obese. So there are neurons that you can control with that precision, where you specifically activate one behavior and not others because you have this precision. And so the question is, if you have a neuron that is damaged because of certain like cell, maybe it is damaged because of some missing receptors, you can go and activate those neurons artificially by artificially expressing these light sensitive channels on those neurons. So even if those neurons are naturally not active, you can artificially activate them. But the road from there to how you cure a disease is still kind of unknown. But the fact that you have such specific control over small neurons give us hope that maybe one day we will be able to perturb like one small part of the brain without affecting the others, because a lot of a lot of the ways how psychiatric drugs work is it just numbs the whole brain, but we can't really go to one part of the brain and cure it or like activate or inhibit it. But it is probably going to be possible in some future, because we have at least some evidence that is possible in a fly or a mouse.
Sure.
Yes, ma'am. Just one moment.
Yes, yes.
Actually, it is better that if you can give the names of the people who have asked this question. That will also be very helpful.
Okay, I don't have the names. These questions are sent
So,
by some student.
whenever you are providing the questions, please also give the names of the people who have asked this question. And by the way, Rajyasree, can I ask one small question?
Mm-hmm.
Actually, I was just wondering that we have this vision and the functional vision and the psyche. That is the multiple centers of the brain, they control these aspects. Normal vision, then psyche, and that is say for example, a particular scene which makes us frightened. So these are multiple aspects of this vision. Are they present in Drosophila?
So it's an interesting question. So so the LC16 neurons are, for example, the neurons that I found that we found in our paper to be to act via the moonwalker neurons to control backward walking. They only respond to slow moving shadows, where there are other neurons that respond to fast moving shadows, which triggers a different behavioral response. There are other visual neurons that respond to different kind of loom or shadow, and results in flight, like not backward walking retreat, but like escape, a different mode of escape. So it seems that even in the fly brain, you have these different classifications of different kinds of fear. Like this is my interpretation, where they can kind of classify what is the nature of the threat, and then choose the appropriate motor action. For example, when a fly is in presence of a praying mantis, the most optimal response is to retreat by backing up. Whereas if it is in presence of a flying dragonfly, it might just fly away. If it was not restricted in a petri dish, it might choose to fly away. So there are definitely, so it's definitely not as developed as higher organisms in terms of what you're asking, but I think there is some primitive blocks of classifying different visual categories of different visual threats, or like different visual scenes. And this neuron is a little more it's not in the it's not on the periphery, it's a little bit central. So it still is a few neurons downstream of the retina or the other neurons that actually so it actually has already processed some information. It's not only it's just not seeing, it's also thinking in a way, you know what I mean?
Yeah, I understand. Thank you. I understand. It will develop to this respect as evolution occurs with phylogenetic development. This will. I understand. Thank you. Over to Case, ma'am.
The next question is from Riya Chakraborty. She is asking that if LC16 and moonwalker neurons are not in close proximity, then how is the information relayed between them?
Yeah, so we haven't found the intervening neuron. So it's usually, say, neuron X or in between LC16 and moonwalker neurons, which probably relays this information from LC16s to the moonwalker neurons. So we haven't found those neurons in between. So that's like still ongoing work.
Okay. Rotish Goswami, he's asking, what is the full form of LC16, and is the reflex action in the human body similar to the moonwalker neurons?
Mm-hmm. So the full form is lobula columnar 16 cells, which is just a part of the visual system in flies. Yeah, we don't know how similar backward walking between humans and flies are, like, in terms of the circuit. So there's always the question of whatever we learn in the fly, whether it's just true for the fly or whether we can extrapolate it to humans. Because, there's always the question of convergent evolution, and so flight developed in different times in evolution. Bats can fly, and dinosaurs could fly, and birds can fly, but all of them either it's not they didn't develop from each other. They kind of found out their own way. So it's an interesting question as to which often challenges how much you learn. But in neuroscience, it's a very challenging thing, because you don't know whether each species figured out its own way of doing a behavior by kind of wiring up the circuits, or whether there is like there is evolution from there are commonalities between species. So it's a good question, and this is like a common challenge in neuroscience.
The last question, it is from Moktan Yukta. The question is, are moonwalker neurons present in other insects or organisms as well?
That's a good question. So we have some neurons in crickets that kind of look like the moonwalker neurons, but we can't ever tell because neurons kind of look very similar. But they also respond to touch and kind of generate a startle response. So we don't know if that is the the moonwalker kind of like a duplicate in another insect. But stick insects walk backwards, and there are many insects which ants walk backwards for long distances. So there has to be neurons that control these backward movement. We just don't know whether they look similarly or whether the circuits are kind of similar between fruit flies and all these other animals, because ants can move for very long distances backwards, whereas fruit flies only walk a few steps unless they're like put into these very narrow grooves. So where the flexibility of backward walking differs insect to insect, so we don't know how much the circuits are conserved and how much it is kind of specialized.
Okay, one more question has come, that is it is asked by Jyotipriya Sarkar. Even if light-sensitive channels are introduced, how can the neurons even function if they are already damaged?
Yeah, so it depends on how. So one thing when we are expressing these light-sensitive channels in neurons, we are not damaging the cells. It is just it's kind of on the membrane of the cells, and they don't really kill the cells unless you overexpress these channels to an extent that the cell then dies. But so if you, I think the question is, if you already have a cell that is like not functioning, whether you can express these neurons, these channels, and make them function. Now that depends on how damaged the cells are. If it's like it depends experiment to experiment. Like it depends on the experiment and what kind of manipulation you're doing, like what kind of damage there is and how much of activation you are kind of causing, and what is the balance between this excitation and the damage. But yeah, it's still not known or how much it can actually circumvent a damaged cell, like the artificial activation, how much it can cure, like how much it can circumvent a damaged circuit.
Okay, do we have time for question answer? Sir?
Yes, ma'am, you can ask. Yes, ma'am, you can ask one more.
So one more question. It is from Shomodi Bhumi. If we can learn more about the circuit of these organisms, will there be a way where we can control other general behaviors as well?
In other organisms or in?
Most probably. I mean that.
Yeah. So I think it's just there are some model organisms that people work with, and they are the favorite model organisms. And people have made a lot of transgenic animals for those model organisms. So a lot of the experiments that are possible in Drosophila is not possible in other organisms, just because they don't have the tools. But with CRISPR and everything, we have come to a point where we can introduce these tools very easily in other organisms, and you can then manage, you can then control the nervous system of other non-model organisms much uh, it might not take as long as it took for us to develop these tools for the model organisms. Because of the technologies that we have right now, we can transfer. So yeah, I think we're coming to a point where we won't be restricted to just the model organisms we work with, but we will be able to manipulate circuits in other organisms like cockroaches, or even cuttlefish, which and all kinds of organisms.
So do you have any wish to shift your model organism in future?
So I have actually switched to mice. So now I'm working on social memories in mice,
Yeah.
in my postdoc. So I switched, yeah, because it was like I'm interested in memory and forgetting and social hierarchies, and so I have moved to mice. But the, so I'm actually very interested in social behaviors. And then there is a there is a vole species called prairie voles, and they are not genetic model organisms, so they don't have many tools as much as mice and fruit flies do. But they have this very interesting behavior where they form like pair bonds. So the male and the female are in love almost, and they live together throughout their lives, which is very rare, which is not true in mice. And then there is a sister species of these voles called the meadow voles, and they are very promiscuous. So the prairie voles are monogamous, and the meadow voles are promiscuous, and they are like just one branch away in the evolutionary tree. And so I'm very interested in how monogamy is represented in the brain, and how like promiscuity is represented in the brain. And then in the future, if I ever have to if I could do it, could address that question, that would be very interesting. Although we don't have the tools to address this now, but maybe in the future there would be a possibility to kind of uh, do that kind of experiment. So it would be interesting to see if I can go to the brain of a prairie vole and make it a meadow vole, turn it from monogamous to promiscuous, and vice versa, because then I will be able to understand which circuits cause this. So yeah, that's definitely
Very interesting.
yeah.
So do you want to say anything to our students to inspire them?
Yeah, I think this is such a wonderful, microbiology. The department is just wonderful, and everybody is. I think just work. I did my first research project in the Department of Microbiology, and that kind of instilled in me this curiosity about not just microbiology, but other fields of biology. And I think that was just amazing. And you're just in such a good place. There's so much exposure that I don't think anything can go wrong if you are like studying in microbiology department in Xavier's. It's like a very special place, because it's not just. I think there was a personal touch to my experience. My experience in this department was like not just academic, but very personal. And I think it just created this culture of sharing. Like we are still friends. Like my batchmates and I were still friends. We still kind of help each other in our careers. So this is just such a great place to be. So, and I think the only thing I can say is, yeah, just don't get too hung up on career plans, because you have to leave some room for luck. But when luck comes, you should be prepared. And I think if you're in this department, you will be prepared. Yeah. Yeah. Thank you.
I also, I also tell you something, Rajeshri, wonderful work. Okay, and we are very much happy that you are sharing your work here, and as a speaker, you are delivering lecture in modern trends in microbiology.
Thank you.
Wonderful,
It's a
wonderful.
pleasure.
I want to congratulate you. Thank you.
Thank you.
Thank you so much.
Thank you so much. Now, as a vote of thanks for her amazing talk, I would request Professor Dr. Kasturi Sarkar to present a virtual bouquet, a memento, and an e-certificate to Dr. Rajoshri Sen.
Oh, thank you so much. Oh, thank you so much.
Surely we'll meet in near future.
Yeah, thanks.
Next time when you come to India, please visit your
Yes, of course, of course.
apartment. Yeah.
I will have another story about
Yes.
mice then.
So actually, Rajyasree, this completes the circle. The circle
Yes.
is completed. You were a student, and now you came back as a guest speaker, honored guest speaker. We are really proud and privileged, and may you actually reach newer heights in future. Thank you very much.
Do more good research.
Thanks.
Do whatever you want to do. Yes. Thank you so much.
Yeah.
This brings us to the end of technical session 1. We shall now proceed to the second technical session of today, presentation by BSc second year of our department. The judges for this session are Professor Dr. Kasturi Sarkar and Professor Dr. Joydeep Ghosh. The time limit for each presentation by each group is 8 minutes and 2 minutes is for question answer session. The first bell will ring at 8 minutes sharp, the second bell will ring at 9 minutes, and beyond that you will be interrupted by the judges. However, in case of technical glitches, you will get extra time. The first group that will be presenting is Sharanya Kundu and Kishore Roy.
Before they present, just one point to mention. I really will appreciate that if you circulate the eDNA to all the participants and the registrants, please kindly do that so that they also have an idea that what are the papers and what topics will be presented today and tomorrow and day after tomorrow. Thank you.
Just a small change. The judges for this session are Professor Dr. Sudeshna Sham Chaudhuri and Professor Dr. Joydeep Ghosh.
When chemistry meets biology, magic happens. A very good evening to one and all present here. Our topic for today is cyborg bacteria. The rate at which the plants photosynthesize food and fuel, that is carbohydrates, with the help of chlorophyll, carbon dioxide, along with sunlight, has sufficed for the past 3 billion years since the evolution of cyanobacteria. But in recent 100 years, it is seen that the plants struggle to feed our exponentially growing human population. And chlorophyll's inability to absorb a large amount of sunlight is perhaps the root cause. At best, the chlorophyll only absorbs 12% of the solar energy, and plants depicts nearly 1% of it. On the contrary, the modern day solar panel have an efficiency of around 15 to 20% only. In 2017, the scientists came up with a much more economic alternative: artificial photosynthesis. Dr. Sakimoto et al. Developed a genetically engineered cyborg bacteria. The term cyborg applies to an organism that has restored function or enhanced abilities due to the incorporation of certain artificial components, which increases its efficiency manyfold. This bacteria, as a natural part of their defense mechanism, selectively absorbs heavy metals and sulfides from their environment to produce metal sulfide complexes. For example, we can have cadmium sulfide, lead sulfide, mercury sulfide, which prevents the heavy metals from spreading into their body, localizing them and preventing heavy metal toxicity in them. These sulfide crystals also have a photovoltaic property, which can act as semiconductors and absorb solar energy, becoming supercharged. These bacteria also, as a natural part of the respiratory mechanism, converts carbon dioxide and the harnessed solar energy to acetic acid, which can then be used for various other industry processes. For example, synthesis of food in preservation industries, that is to increase the shelf life of our food, fuels, plastics like polyhydroxybutyrate, and various other commodity chemicals. Now, over to Kishal for the rest of the tickets.
Thank you, Sharanya. Now, for the synthesis of cyborg bacteria, Dr. Sakimoto chose the bacterium Morella thermoacetica because, firstly, Morella thermoacetica is an anaerobic, non-photosynthetic, astrogenic bacteria which reduces carbon dioxide to acetate. And secondly, it forms heavy metal sulfides when exposed to heavy metal containing environment. So what they did was, as a part of their experiment, they kept large culture vats containing cadmium, containing amino acid cysteine, and the bacterium in sunlight, which resulted in the formation of CDS nanocrystals on their body surface. So these self-genetically engineered Morella thermoacetica CDS, that is the cyborg bacteria, could absorb solar energy with an efficiency of about 80%. Moreover, it's a zero waste process and is self-replicating and self-regenerating because only one-time incorporation of source of cadmium and cysteine is required. Then it gets multiplied many folds with each bacterial cell division, thereby making this process very efficient and cheaper when compared to solar panels. As there has been an increasing demand of fossil fuel because of increasing population, this harnessed solar energy can be an alternative clean source of energy to meet people's demand, like for generation of electricity. It acts as a natural sink for carbon dioxide because this carbon dioxide gets reduced to acetic acid, which can further be used for different industrial processes. Heavy metals like cadmium, mercury, lead that causes environmental toxicity and adverse effects in animals and humans like chemical poisoning, on the contrary, can be used to harness solar energy in much more effective manner. And lastly, these large number of culture vats can be accommodated in a smaller space. With advantages, scientists also found some drawbacks of the experiments. Firstly, they found out that electron in the cadmium gets excited when as soon as the sunlight falls on it. So these were lost through the interaction with other bacterial cell components. And secondly, scientists as cadmium is very harmful and hazardous for the person, as well as environment if proper precautions are not taken. So scientists are trying to find out a less harmful source compared to cadmium to work with. Now, as this process is 6 times more efficient than natural photosynthesis and 4 times more efficient when compared to commercial solar cells. The scientists are working on this and trying to understand the mechanism by which the cadmium is taken up by the bacteria and they form ECM crystals. And they are also trying to make this process more efficient and safe so that its use can be commercialized. Now, on weighing the pro side and con side, we get to see that pro side of the beam balance is much more heavier. Thus, this process can prove to be a boon for developing countries. Thank you.
Very nice presentation. Ishal and Sharonna, can you please show me that slide? Just I want that sulfide drill, the second one, second slide.
This one.
Uh,
Ma'am, which one?
the second one. This is the title slide. Yes, yes. No. Then the sulfide drill containing thing.
Ma'am, this.
Yes. I just want to see. Can you please once again describe this one? So,
Ma'am, actually, these bacteria, as a natural part of the defense mechanism, can selective from the environment, immediate environment, they can selectively absorb cadmium and sulfide, so they can prevent the spread of this cadmium into their body by localizing them by forming cadmium sulfide crystals. So the cadmium get arrested and therefore it cannot harm their body. It cannot cause a toxicity in them. And this in turn can act as photovoltaic cells, or we can say that they can act as semiconductors, so that when these semiconductors are energized with solar energy, they can produce energy, electrical energy rather, with a much more efficiency of 80%, because these bacteria can multiply on their own. So we can get a large amount of cadmium sulfide crystals in this way.
okay, thank you, Sharonna. Wonderful. That's all.
Thank you, ma'am. Thank you, ma'am.
I don't need to ask anything. Thank you so much. Well explained. Very well explained.
Thank you, ma'am.
Thank you, ma'am. Thank you, ma'am.
This
Ma'am,
is
can I
JG.
stop?
Yes, sir. Yes, sir.
Yeah, I just want to ask a small question. So it is true that this bacteria is accumulating the toxic cadmium and other metals, and thereby it has a big impact in the environment. But for all these type of bacteria, there is one basic problem. They are accumulating, thereby generating very high concentration of metal ions within them. But all bacteria, each bacterial cell has a definite lifetime. It cannot live forever. Some of the cells, whatever application you put the cells in, some of the bacterial cells will die after some time. Then more bacteria in your application will die, and after death you have cell lysis, and then the toxic metals will again come out.
So it will remain it will remain confined to the culture broth in the vats. vats only, but we have to take proper precautions to dispose it. Some when
Important to
cadmium.
dispose it.
Yeah. So, what I wanted to ask you is that can you think of a strategy
So
to take
if so if
to
the
take it out?
cadmium perhaps reaches the soil, so we can do one thing. We can perhaps incorporate cellulose particles or wood particles in them. So the wood particles will perhaps absorb rather than absorbing the cadmium crystals. They will absorb it, and the cadmium crystals will bind to it, and thereby the roots of the plants won't take it up, and it would be rendered inactive. Or we can do one thing, we can just prevent the spreading. Even if we don't do this, we can do the bioremediation by phytostabilization. That is, we can stop soil erosion, or we can just prevent the mobility of these heavy metals in the soil by some means.
Okay. Yeah. So you've got some ideas, which seems quite logical. So thanks to both of you.
Thank
Good
you, sir.
presentation. Yeah.
Thank you, sir.
Thank you, Sharanya and Kishore. The next group that will be presenting is Ayush Bagchi and Stuti Barua.
On 12th March 2016, strange blades of ice were found on the Mount Uyayako, the second highest volcano. These unusual formations are called the Nevis penitents, were found to be hosts to a community of microscopic life. So this was the first time life had ever been recorded in these icy pinnacles. It was an important discovery as it could provide an insight on extraterrestrial life, as these formations were also believed to exist in other planetary bodies, including Jupiter's moon Europa. Ladies and gentlemen, I, Stuti Barwa, along with my partner Ayush Bakshi, would like to give an illustration on what the penitents are and how it could lead it to a quantum leap in the study of alien life. Okay, so what exactly is the Nevis penitents? So they are these big, tall, pointed structures made of ice and are found in clusters. They are found in high altitudes in snow-covered areas, which is around 4,000 meters. So these penitents now require a very unique combination of UV radiations, low humidity, excessive dry winds, and fluctuating temperatures. So what happens is when sunlight falls on these icy surfaces, it causes sublimation. And due to the uneven distribution of the sun rays, there are regions which sublimate more faster than other regions. And the sublimation causes formation of depressions, which then concentrate more sunlight, speeding up the process of sublimation and leaves behind these assemblage of the beautiful crystal-like ice blades. Okay, so now here is a picture of a man along with the penitents, just to give a rough idea as to how tall they can grow. So the penitents can now be like from few centimeters to like very high, up to 5 meters. Now, to come to the importance of it, these structures have been reported on Jupiter's moon, Europa. So with the discovery of snow algae on these structures, there might be a possibility of life found in them, which could contribute immensely to the field of astrobiology. So now Ayush would like to elaborate on the research involved under penitents.
Mount Uyayako, present near the Atacama Desert in Chile, is one of the driest places on Earth. It has a similar environment to Mars. Dr. Stephen Pointing, a professor of microbial ecology at Yale University, says that if a bacteria was transferred from the Atacama Desert to Mars, it would be able to survive there. Now moving on to the discoveries made by Lara Vimersati and co. A total of 7 ice samples were collected by scraping the sides of these ice structures found on a penitent field on Mount Uyayako, 2 from clear ice and 5 from the visible red patches of ice. Standard procedures of DNA extraction, polymerase chain reaction, and sequencing were performed. After using a bioinformatics package, the top operational taxonomical units, or OTUs, were verified with the BLAST tool to search the NCBI database. As you can see on the graphs on the right, maximum number of OTUs were observed in the soil in and around the penitents for both eukaryotes as well as prokaryotes. However, if you follow the red line carefully, you'll find that there were a significant number of OTUs present in the red ice samples. Around 75 to 80% of these belong to a category known as snow algae if you follow the topmost histogram on the right. Red eye samples were dominated by a particular algae, Chlamydomonas nivalis, which had an absolute match with the NCBI database. Now, Stuti is going to talk about this particular algae.
Chlamydomonas nivalis is a unicellular red-colored photosynthetic green algae that is responsible for the phenomena of watermelon snow, where portions of ice appear pink or red. So this color is due to the presence of a secondary carotenoid called the astaxanthin. So now Chlamydomonas nivalis is found in some of the most life-limiting regions of the Earth. They are called cryobiomes, that is, they find optimal living conditions at very low temperatures, which ranges around 0 degrees Celsius. So anything above 10 degrees is lethal for them. Also, snow algae can often be found in association with bacteria, fungi, and other viruses, ciliates. And there has been some instances where gram-negative bacteria has also been reported. So now, to come to the most important thing is, how do these snow algae really adapt in such low temperatures? So there are many generalizations to it. So first is, these plants protect the protoplast by undergoing gradual gelation, and that prevents the deformation of the cells. So while this granulation occurs, there is increase in the permeability of the cell membranes, which facilitates the migration of the cell sap during overcooling and prevents the formation of ice crystals in the cells. Thirdly, thick cell walls also efficiently protect the cell from the really cold temperatures. And lastly is, microscopic examinations of this red eye samples have revealed the presence of cysts or spores, which are the resting or the dormant stage of the microorganism, which helps it survive the unfavorable conditions. So now here are some pictures of snow watermelon, like how watermelon snow actually looks like. So they are taken from different Arctic and alpine regions. And no, they don't taste good, in case you're wondering. Okay, now Ayush is going to take you on a journey through the cosmos, millions of light years away, to Jupiter's moon Europa.
So we've given these pictures on a screen just to give everyone a rough idea as to what Jupiter's moon Europa actually looks like. As you can see, it is roughly the same size as our moon, which is important to note, as you can only imagine how small it is compared to the massive gas giant Jupiter. The next picture is just a closer look at Europa's surface. The red color is due to the presence of sea salts and minerals. And the last picture isn't really important, but it's just to give you a rough idea as to how far we've come from the year 1972, that is when this picture was taken, and to the present day in 2020. Now, Stuti has already talked about what penitents are and how they are formed. Now, imagine these penitents, but 3 to 4 times larger. Europe is a much colder, a much drier climate than Earth, and it's a thin atmosphere. So penitents can reach up to a height of 15 meters. That's almost the size of a modern-day 4-story building. Apart from the possible presence of penitents, Europe has clearly visible lineae on its surface. These lineae are believed to have similar origins as oceanic ridges on Earth. As you can see, it's as if the crust is divided into blocks. So now I'm going to talk about the most exciting part of this presentation, the presence of an ocean below this icy crust of Europa. The presence of lineae is already an indication that there is water activity below the surface. On top of that, the absence of extreme structures such as mountains also supports this fact. On top of that, Jupiter's immense gravitational pull causes the generation of heat energy in Europa's core, enough to maintain a liquid ocean. To give you a rough idea as to how strong this gravitational pull is, the gravitational pull between Earth and the moon causes tidal waves on Earth. So one can only imagine how strong the gravitational pull of a planet 318 times the size of our own has. The possibility of a subsurface ocean was further clarified by observations made by the Hubble Space Telescope in 2016. Water vapor plumes were found to come out from the bottom left of the planet as indicated by the arrow. This would mean that there are presence of ice fractures on the surface, which could form a protective habitat. So these ice fractures could harbor life such as snow algae, as we've previously discussed. These ice fractures are in close proximity to the water and salt from the ocean and to the sunlight coming from the sun. So if we give more thought to it, we find striking similarities between the conditions on Earth during the origin of life and conditions on Europa. So NASA's Europa Clipper is set to launch in the year 2024. It'll be the first mission to deal solely with Europa. Its mission is to investigate the habitability of Europa and to find any future landing spots for future missions. So in summary, Europa may have enough water, may have enough energy being inner heat energy and external solar radiation, and chemicals for the beginning of an abiogenesis completely different from our own.
In conclusion, we think any of these could be found on Europa or in Europa's ocean. Of course, any life found should be primitive. So at least according to our present knowledge of life on Earth, we can only propose the presence of these microorganisms and not higher life forms. So these are extremophiles. Now we have, if you see, we have given extremophiles in double quotes because an extremophile on Earth isn't an extremophile on a celestial body where these extreme conditions are quite frankly normal. Thank you, judges and audience, for your patience. We hope you enjoyed our presentation.
They can tolerate temperature, but how they will tolerate that intense pressure? How they become barophilic? What are these adaptations that make them barophilic? Can you answer?
You wanna go? No, like sir.
Sir, barophilic. So the thing is, sir, we have just predicted that there might be barophiles because of the presence of ocean. So we think there might be barophiles because of the high pressure, because of the presence of ocean.
And how they actually can withstand that high pressure? How normally they do that?
Yes, sir. To add on to that, our major point was that life will probably be found on the ice fractures that's near to the surface, not closer to the bottom of the seabed. Like also because of the water vapor plumes, possibility of deep sea hydrothermal vents can be there, just like on Earth. So as we know that there are complete ecosystems around these deep sea hydrothermal vents. So similar organisms can be found, sir. But our most promising is the ice fractures because it is a protective environment which has energy sources from both the sun as well as the liquid water from below, which has sea salt.
Okay.
Was I clear?
Chlamydomonas nivalis is a green algae, but it appears red because of carotenoid. But carotenoid, they are orange in color. How the red color is coming?
So it is because it matures. It matures over the time, sir. So because it matures, and due to the ice present, it becomes pink. It is these orange. They are first present as green spores, as biflagellated spores. So as the time goes on, as nutrients deplete, and as they are stagnant in that place, that's how they gradually become darker in color. And yes, they become darker in color like that. The secondary carotenoid accumulates in the cells. Sir.
Has it got anything to do with the dead cells? The pigments are not getting decomposed and they are deposited on the surface. Anything to do with that?
Sir,
because algae cannot be perennial. It cannot be perennial though.
It's not only dead cells, it's even during extreme, like during winter when the temperatures are much lower. So they gradually move to spore formation in order to withstand this temperature. So spores are staying there for a long time. So gradual maturation of the red color to the red color occurs.
Okay, thank you.
Thank you, sir.
Thank you. Very nice presentation. I don't have any questions to ask. Joydeep sir may ask, but one question. Can you please show me that blast analysis part where the similarities are there with both eukaryotes as well as prokaryotes? Can you please just show me that slide? Yes. So yes, that particular slide. So what you have told me, yes, O2, right? O2 is operational taxonomic unit. Okay, just let me see this slide. The alpha refraction comes based on the number of. Okay. So O2 is taken as for what? For what sort of work here?
Okay, so basically, we're taking samples from both clear ice and red ice just to make sure that if there's life on the red ice, that it's not there in the clear ice. Like red ice is particularly unique. So after extracting the bacteria, like the microorganisms, we're doing DNA extraction and we basically find the sequences of the microorganisms that we find. That is like these are the possible sequences. Like some of them have 100% match with the database, while some of them are close strains to the ones that are already discovered. Like 96% doesn't mean that it's actually quite far from the strain. So
Yes, and 100% with the
Yes.
chlamydomonas.
If you want to see this, like, Chlamydomonas
Yes.
was the only dominant species which had absolute match. Yes. And the closest one was
Very well described and very novel thing. Very nice. Thank you.
Thank you, ma'am.
Thank you, ma'am.
Yeah, I just have one small question.
Yes, sir.
These microorganisms which survive and grow in these extremely cold environments, they have a certain, they actually overexpress a certain group of proteins that protect them from this extremely low temperature. Do you know what are these proteins called?
Sir, antifreeze. Oh, wait, you can't show. Oh, you can. Sir, are they antifreeze proteins?
Yes, one type of protein is
Yes, they're-
called antifreeze proteins.
They're
Yeah,
also
I know.
found. They're also found in like higher organisms. For example, fish that survive in these cold climates. They also have antifreeze proteins. So that's something common
Exactly.
between microorganisms and fish.
Exactly. And
Um,
a more general class of proteins which are particularly expressed when this temperature becomes too low. As one type is definitely antifreeze proteins, and the other ones, yes.
So I don't. So I don't know. Suki, if you.
Not sure, sir.
Yeah, they are called just like heat shock. These are called cold shock proteins.
Okay.
Cold shock proteins. Yeah.
Okay.
Thank you, sir.
Thank you, sir.
Yeah. Thank you very much. Very nice topic. Very nice presentation.
Thank you, sir.
Okay. Yeah.
Very nice information about the cold shock protein. I just want to ask you one thing as you were mentioning about the antifreeze protein. Can you please tell me the nature of such antifreeze protein and um, how far they are acting as antifreeze protein? It's obviously the survival strategy of many of the archaebacteria as well as many other organisms which actually endures very low temperature. So can you please tell me what is the nature of antifreeze protein? So that is the name of the antifreeze protein actually. Any idea what sort of proteins they are?
Is it because they're prevalently hydrophobic in order to not be in contact with the water?
They are not hydrophobic. They are all glycoproteins. Okay. Yes, glycoproteins. So as they are glycoproteins in nature, they actually gives a type of slippery basis. So that's why the ice crystal cannot grow over them. So as they are glycoprotein in nature, the sugars they actually renders a kind of slippery base. So at that particular base, ice crystal cannot grow. Thank you.
Thank you, ma'am.
Thank you, Ayush and Stuti. Next up, we have Ritushri Ganguly and Vaibhavi Bhattacharya.
Good evening, all the judges and the listeners. This is Vaibhavi Bhattacharya.
And this is Ritushri Ganguly.
We are going to present our study about bioluminescent bacteria, a pollution detector in economic and eco-friendly way. With this increasing rate of industrialization, there has been huge rate of effluent discharge into the water body. This has led to eutrophication and heavy metal pollution. Also, there are cases of antibiotic toxicity which hampers the marine life. That's why it is very important to keep a regular check on the toxicity concentration of these pollutants. But that process is practically not feasible because it is expensive, time consuming, it requires sophisticated gadgets and highly skilled manpower. That's why we have come up with an easy solution, which will be further discussed by my partner, so over to her.
Good evening, everyone. In this presentation, we will be dealing with bioluminescence assay. Now, this method helps in an early detection of eutrophication, that is way before red tide. It enables an easy monitoring of heavy metal toxicity in the water bodies. Besides, the method is rapid, cheap, and simple, thereby making it economic as well as eco-friendly. Now, before proceeding further, let us know what bioluminescence is. In layman's terms, it refers to the production of light for defense, predation, and mating. Coming to the enzymology part, bioluminescence is brought about by a cascade of chemical reactions under the influence of the enzyme luciferase and lux operon, which comprises of a set of 8 genes. As shown in the figure given alongside, Lux AB luciferase brings about the monooxygenation reaction, thereby producing light at around 490 nanometer from an excited state flavin 4A hydroxide. Lux CDE constitute the fatty acid reductase complex, which helps in the synthesis of long-chain aldehydes, while Lux G codes for flavin reductase, which helps in the regeneration reaction as shown in the slide. Now, bioluminescence is usually seen in the stationary phase of the bacterial growth. There are basically 4 classes of bacteria which show this phenomena. These are Vibrio, Photobacterium, Shivanella, and Photorhabdus. Moving on to our next slide, wherein we will be discussing about our experiment for the detection of eutrophication, which in simple words is characterized by an excessive growth of plant and algae in the water bodies. Moreover, the usual concentrations of nitrates and phosphates to cause eutrophication are 0.2 mg per liter and 0.02 mg per liter respectively. As for the experiment, the bacteria which we have studied is Photobacterium phosphorium, and the principle involved in this experiment is a change in concentration which in turn affects the luminosity. Therefore, as shown in the 3 pictures in this slide, various concentrations of nitrates and phosphates have been added to Photobacterium phosphorium cultures designated as A, B, C, and D. It is seen that as the concentration increases, the luminosity decreases. For potassium nitrate at around 0.1 mg per liter, the luminosity decreases drastically till it disappears around 0.2 mg per liter. Similar is the case for ferrous nitrate. For phosphates, between 0.01 and 0.02 mg per liter, the luminosity is found to be minimum. However, in natural scenario, since nitrates and phosphates are present together during eutrophication, another study has been conducted wherein various concentrations of phosphates and nitrates have been added in combination to A, B, C, and D. Here it is seen that even before the concentration required for eutrophication is achieved, a drastic decrease in luminosity is observed, as seen in case C. Therefore, we can conclude that Photobacterium phosphorium can be used as an indicator to detect eutrophication way before red tide. Now over to you, Vaibhavi.
Now, for understanding the case of heavy metal pollution, we have taken up hexavalent chromium ion, which is a potent genomic carcinogen. The bacteria studied in this case is Vibrio campbelli, which is isolated from the gut of sea urchin, and the colonies were cultured on TCBS media. We did the general enzyme profiling of the bacteria, and it revealed that it can produce all these enzymes, making the bacteria industrially beneficial. Next, we will understand the growth kinetics of the bacteria under chromium-6 stress. For that, we have plotted a graph taking the optical density value at 260 nanometer and have compared the control setup with all other concentrations ranging till 12 ppm. It's found that for 2 and 4 ppm, there is not much deviation from the control setup, but for 8, 10, and 12 ppm, there is a reduction in the stationary phase, as well as the decline phase. It starts early. As already mentioned, that bioluminescence happens only during the stationary phase. That's why there is a reduction in the bioluminescence, which is visible to the naked eyes only up to the concentration of 8 ppm of chromium-6. Next, we are putting these 2 case studies and the principle, that is decreasing luminous intensity, to develop a prototype biosensor. Herein, the bacteria of interest may be Photobacterium or the Vibrio species. They are cultured in their respective media, and agar cubes are cut out. These cubes are packed in highly perforated poly packets, which are then placed in flask which contains the water sample to be tested. Then we incubated it and finally got the result that there is a decrease in luminous intensity with the increase in pollution concentration. And
This principle can be applied in future to make a software which can take up the intensity value and give us the approximate concentration of the pollutants. That will make this whole process a faster and a more precise one. Now, I would like to compile the advantages of using this biosensor. The first one, it can be handled with only basic laboratory skills. There are no complex technicalities involved like that of a transducer or any microelectronic devices. And as already mentioned, the radiations are from the visible range. Therefore, there is no question of any harmful radiation and the work environment is safe. And finally, since we are only using bacteria and no external cell culture, that's why that reduces the cost as well as makes the process ethical. And these all reasons come up together to help us keep a check on the concentration of the toxic substances regularly, thus enabling us to take necessary steps whenever required. Now, this principle can be extended to check the concentration of various other drugs by studying the growth kinetics of Vibrio fischeri, also for checking the adult concentration in food microbiology assay, and for checking the efficacy of virucides without the help of an external cell culture. So on a conclusion note, I want to say that this biosensor is of great advantage to nature because it is simple, it is easy to use, it saves time, it saves electricity, and I believe that's why it should have a great scope.
Now, we would like to acknowledge our referential sources, the first two being our main papers and the last two being the extensions. The images which we have used in our presentation have also been taken from these papers. With this, we come to the end of our presentation. Hope you all stay healthy amidst these tough times. We would also like to express our gratitude to our listeners and our professors for giving us this wonderful opportunity. Thank you so much and do stay tuned.
Very well organized and wonderful presentation.
Thank you, ma'am.
Can I ask you something?
Yes, ma'am. Okay. Sure, ma'am.
So in your slide that you have told that this particular thing can be used for drug assay.
Yes,
can you design an experiment? Can you propose how far? Yes.
Yes, ma'am. I'm sure.
The various drugs can be checked.
Yes, ma'am. Ma'am, there was a paper that this paper deals with it basically. Here, the principle lies the same, that as the concentration of this antibiotics, it increases in the water bodies. So we are taking up that sample and we are using Vibrio fischeri. Now, if there is a high concentration of these antibiotics, then Vibrio fischeri won't be able to survive. And if there is a lower concentration, then it will be surviving and the intensity, that is 490 nanometer of blue-green light, will be visible. And not only for antibiotics, it can be done for antihistamines, anti-inflammatory drugs, anti-steroidal drugs as well.
Very nice answer, Vaibhavi. Very nice. I just want to add something here. So students, in your last semester, you will have some critical review type of thing, right? So the thing that you have done already, it's a kind of that kind of approach. So you are not only going through one particular paper or not only one particular case study, but you are also consulting the cross references. Okay, so that is the main motto of this your critical review or the or reviewing any particular paper or review any particular topic.
Ma'am, actually, we took up the topic of bioluminescence and how we can use it to develop a biosensor. So these are the cases we found, like for drug assay, for food microbiology, and heavy metals. So that was
very nice homework and cross-reference study, etc. Okay, thank you.
Thank you, ma'am. Thank you, ma'am.
I have a question.
Yes, sir.
Can you guys please show me the slide where you showed the reaction of Lux CD complex?
Yes, sir. Sure, sir.
Yes, this one. So here you see that the Lux CD complex is actually a multi-enzyme complex, right?
Yes, sir.
So in nature, in every like cell, prokaryote to eukaryote, they frequently use these type of multi-enzyme complex. There are many reactions in normal metabolism and the special cases like this one. So I just want to ask you a very basic question. So what is the major advantage of this strategy of using a multi-enzyme complex compared to separate subunits? Imagine that the same reaction could be performed if the subunits were separate, not as a complex. But forming a complex has a definite advantage over separate units. What is that?
Sir, what I think is like because this is like the proteins, they are synthesized after transcription and translation. So if there is only one promoter, after synthesizing from that one promoter only, we are getting all these subunits together. But had it been in different subunits, like not a multi-complex, then there would be many promoters involved. So in that case, like maybe due to some disorders, if one promoter is blocked, if on one promoter the RNA polymerase is not able to act, then maybe it's not working.
I understand what you say. Yeah, but see, there are many other. So you are talking about the concept of, say, operon. So
Yes,
one
sir.
promoter regulating many genes.
Yes, sir.
So this operon concept is mostly applicable to prokaryotes. First of all, it is not that common in eukaryotes. But but there are many examples where operons are encoding different protein. One operon definitely encoding different proteins, but they are not acting as multi-enzyme. They are separate, like say lac operon or any other operon. The enzymes, they are separate. So it is not always true that one promoter expressing many genes will always lead to multi-enzyme complex. No, that is not the case. But the real point here is, see, this multi-enzyme complex is catalyzing more than one reaction. So say there are four reactions. So each reaction will have a product.
Yes, sir.
And if it is a multi-enzyme complex, then the product can easily move from one subunit to the next subunit for the next reaction. Then the second product can go easily to the third subunit for the third reaction. But if the enzymes are separate, then after product 1 is formed, product 1 has to diffuse to the second enzyme. Then you form the product 2, then the product 2 has to diffuse to the third enzyme. So that is much less efficient than a multi-enzyme complex strategy. This is basically to channelize one product to the next enzyme, then the second product to the other enzyme. Very easy. Okay?
Yes, sir.
So thank you. It was a very nice presentation. Yeah.
Thank you, sir.
Thank you, sir.
Thank you so much.
Thank you, Ritushri and Vaibhavi. Next up, we have Ankur Chattopadhyay and Shashwato Ganguly.
A very good evening to everyone present in this webinar. It is indeed an honor for my partner Ankur Chattopadhyay and me, Shashwato Ganguly, to present a paper here. The topic of our today's presentation is cytosolic gram-negative bacteria Shigella flexneri prevent apoptosis by inhibition of effector caspases through lipopolysaccharide. Now moving on to what apoptosis actually is. Now, apoptosis is nothing but a programmed cell death process that eliminates the replicative niche of intracellular pathogens without involving major inflammatory responses. The core of apoptotic machinery consists of enzymes called caspases that exist in the cytoplasm as zymogens or inactive precursor molecules, and they can be broadly subdivided into two categories: initiator caspase consisting of caspase 8 and 9, and the executioner or effector caspase consisting of caspase 3 and 7. In this paper, it is shown that intracellular cytosolic gram-negative bacteria like Shigella flexneri are capable of inhibiting this effector caspase activity and thus successfully blocking apoptosis. The data identified lipopolysaccharide or LPS as a major or bona fide effector caspase inhibitor involving in its O antigen as the main effector caspase binding moiety. Now moving on to the results and discussions, the first point under this is intracellular Shigella blocks apoptosis in epithelial cells by inhibiting effector caspases. Experiments have revealed that neither caspase activity nor apoptosis could be detected in Shigella infected epithelial HeLa or HCT cells. To address this notion, it was examined whether cytochrome C or exogenously added recombinant active caspase 8 and 9 are capable of inducing this effector caspase activity in cytosolic extracts derived from both infected and non-infected cells. It was observed that in case of non-infected cells and cells infected with the non-invasive Shigella BS176 strain, caspase activity was detected and apoptosis occurred. Whereas in case of the cells infected with the invasive Shigella M90T strain, caspase activity was not detected. Here we have three plots demonstrating the caspase 3,7 activity against the exogenously added compounds, that is cytochrome C, active caspase 8, and active caspase 9. The red lines indicate the cells infected with the invasive Shigella M90T strain. The green ones indicate the cells infected with the non-invasive Shigella BS176 strain, and the black ones indicate the non-infected cells. From all the three plots, it is quite prominent that in case of the cells infected with the Shigella M90T strain, there is negligible increase in the effector caspase activity, indicating that apoptosis has been successfully stopped in these cases. Whereas in the other two cases, we can see that there is a considerable increase in the caspase activity, thus indicating apoptosis occurs as normal in those cases. Moving on to the next portion, that is how effector caspases get activated. As we have already spoken, effector caspases are present inside the cell as zymogens or inactive 32 kilodalton precursor molecule. In order to get activated, they have to undergo two successive cleavages. The initial proteolytic cleavage by caspase 8 or 9, generating the P20 subunit, which is an intermediate, followed by the autocatalytic cleavage by caspase 3,7 itself, whereby it removes its own prodomains to ultimately generate the mature P17 subunit of effector caspases. This P17 subunit is observed in non-infected cells and cells infected with non-invasive Shigella BS176 strain. On the other hand, the P20 subunit can be only observed in cells infected with the invasive Shigella M90T strain. Moving on to the next point, that is cytosolic gram-negative bacteria promote caspase inhibition. As we have already seen, effector caspase activity was impaired only by invasive Shigella M90T strain, but not the non-invasive Shigella BS176 strain, and neither by the heat-killed Shigella M90T strain. To further consolidate this observation, a parallel set of experiment was performed using the two separate bacteria, Salmonella enterica serovar typhimurium and Listeria monocytogenes, along with this Shigella M90T. It was observed that both Salmonella and Listeria were unable to impair this effector caspase activity, unlike Shigella M90T. To prove this, here we have provided two Western blot analysis results, whereby we can observe that the P20 subunit can be observed only in cases of the cells infected with the Shigella M90T, whereas in all other cases we can observe the mature P17 subunit. This indicates that apoptosis occurs in all other cases except in case of the cells infected with the Shigella M90T. Now my partner Ankur will continue from here. Over to you.
Thank you, Shashwat. So now we have to discuss about the mechanism of inhibition of effector caspases. So LPS binds and inhibits the effector caspases. LPS or lipopolysaccharide is an essential outer membrane glycolipid that is found in almost all the gram-negative bacteria, and it is also found to initiate certain diverse immunological responses in the host. In addition to this, LPS-induced inflammatory caspase, that is caspase 4, 5, 11, their activation leads to pyroptosis via the production of Gas-Dermin D and plasma membrane rupture without involving canonical inflammasome. Now, pyroptosis is a process which is different from apoptosis. In simple terms, it is a lytic cell death program that is detected by lactate dehydrogenase release. Now, in addition to all these, LPS is also found to inhibit the effector caspase activity in bacterial cell lysate after treatment with TRAIL, the apoptosis inducer, which further proves the anti-apoptotic capacity that is exhibited by LPS. Now, after Western blot analysis, it was already revealed that LPS can successfully inhibit the autocatalytic activation of recombinant caspase 3, whereas the proteolytic activation of caspase 3 from the initiator caspases was unaffected, suggesting that LPS selectively binds and inhibits the effector caspase activity, but not the initiator caspases. So here is a graphical representation showing the relationship between effector caspase activity in bacterial cell lysate in the presence of LPS. From this graph, it is quite clear that there is no significant increase in all the plots representing the caspase 3,7 activity in the bacterial cell lysate in the presence or absence of trail, which further proves that due to the presence of LPS in bacterial cell lysate, it potently inhibits the effector caspase activity. Now, inflammatory caspases exhibit certain specific LPS binding capacity due to the presence of caspase activation and recruitment domain, also known as CARD. Whereas in case of effector caspases, they lack CARD, but they can still efficiently bind to LPS. In addition to this, lipid A, the hydrophobic domain of LPS, is found unable to disrupt the interaction between the biotinylated LPS and the effector caspases, suggesting that lipid A is certainly not the binding moiety of LPS to the effector caspases. In addition to lipid A, LPS also exhibit a non-repeating core oligosaccharide and the distal polysaccharide domain, also known as O antigen. Experimenting with different LPS variants lacking O antigen, it was found that these variants are not able to bind and inhibit the effector caspases, suggesting that O antigen itself is the binding moiety, and this binding is totally independent of lipid A. Coming to our next point of discussion, that is O antigen deficient Shigella strain does not inhibit the effector caspase activity and apoptosis. So in contrast to invasive Shigella M90T strain, the cell death following the Shigella delta galli strain, which is a mutant strain lacking O antigen in its LPS, the infection initiated with a slight activation in the effector caspases along with apoptotic signs in the earlier initial time point of infection that further leads to reduced LDH release as well as decreased intracellular bacterial replication. In an experimental setup consisting of a mouse model, it was found that only Shigella M90T invasive strain, but not the Shigella delta galli strain, caused mortality in the mouse, which further proves that O antigen deficient mutant Shigella delta galli strain does not inhibit effector caspase activity as well as apoptosis due to the fact that O antigen itself is the binding moiety. Now, from all this experimental discussion, we can come into certain conclusions that firstly, apoptosis is very important in controlling the intracellular bacterial colonization. The inhibition of effector caspase enzymatic activity is again very important for ensuring the replicative niche of the gram-negative intracellular pathogen. The inhibition of apoptosis is associated with the gram-negative pathogenic interaction, for example, Shigella flexneri, Rickettsia rickettsii, as well as Francisella tularensis. The chemical composition and the length of the O antigen can be a reliable indicator to the bacterial virulence. As well, the modification of O antigen can help to bypass the host cell defense mechanism. Now, this theory has opened up a new horizon regarding the field of pathogenic interaction to the host cell, and this can be utilized sooner or later in drug therapies whenever the exact mechanism or pathway of pathogenic interaction to the host cell will be known, so that it can be utilized to derive certain specific drugs or technique against certain specific bacterial disease to further ensure human welfare. This is the overview of the entire process we have discussed so far. These are all the references we have used for this presentation. Thank you.
Overview presentation, Jeta Didi, that was very nice because all the mechanism of action is summarized in one slide. Arekbar dekhabe? Can you please show? Fine. I don't have any questions to ask. Thank you.
Thank you, ma'am.
Thank you, ma'am.
Yeah, it was a very good presentation. And I have a very basic question. Say you guys are working in a lab which doesn't have the access to sophisticated cell biology techniques, etc. So your boss is giving you some dead cell samples, some from some cell culture. Your boss is giving you some dead cell samples and asks you that do a simple experiment and tell me whether these dead cells are dead due to apoptosis or necrosis. Necrosis is the normal cell death due to some damage, and apoptosis is the programmed cell death that happens due to some factors. So he is providing you only an agarose gel apparatus, some agarose, some buffers, and ethidium bromide. Basically, the components for agarose gel electrophoresis of DNA. Now, how would you use this simple setup to ensure that the dead cells are dead due to apoptosis and not necrosis? You just have to run an agarose gel, stain it with the Ethereum bromide, and the kind of bands that you get, whatever you get, bands or something else you get, will tell you whether the cells are dying due to apoptosis or necrosis. You see, when a cell die, the components are degraded, particularly the macromolecules are degraded. And you know, all cells have chromosomes, so the chromosomes will be degraded. So in necrosis, there is a special type of DNA degradation pattern. And in apoptosis, there is another type of DNA degradation pattern that could be easily visualized by just just extracting DNA and running the DNA in agar cell. So do you know the difference in pattern or the unique DNA degradation pattern that is observed in from like apoptotic cell when you run agar cell?
No, sir, we are not very sure.
Yeah, so it is when apoptosis happens, the chromosomal DNA is degraded in fragments. So if you run it in agar cell, you will see ladders,
Okay,
distinct ladders. But if it is dead by a necrosis, then the DNA is randomly degraded and you will get a smear. You will not get any ladders. So it is so that's how there is a sort of simplest way to detect apoptosis in cells.
all right, sir. Thank you.
Yeah, so it was very good to listen to your presentation. Thank you very much.
Thank you, sir.
Thank you, Ankur and Shashwato. Next up, we have Srija Kaur and Suparna Das.
Good evening, everybody. I am Srija Kaur from BSc second year Microbiology Department.
Good evening, everyone. I am Suparna Das from BSc second year Microbiology Department. Today we are going to discuss about the role of microorganisms in the biodegradation of synthetic and natural plastic. Plastic disposal is one of the greatest problems facing the environment today, as vast amount of synthetic plastic remain non-degradable. A number of microorganisms like Streptococcus, Aspergillus niger have the ability to degrade different types of plastic under suitable conditions. Now the basic question that arises in our mind is, what are plastic? As we all know that plastic is a synthetic polymer and its component element includes carbon, hydrogen, silicon, oxygen and chloride and nitrogen. It is derived from different sources such as petrochemicals. A key breakthrough came in 1907. Chemist Leo Henrik Beckland, who coined the term plastic, invented the first fully synthetic plastic called bakelite. There are various types of plastic, classified according to their properties such as thermal properties, chemical properties, etc. Based on the thermal properties, plastic can be divided into two groups, thermoplastic and thermosetting polymer. On the application of heat, thermoplastic undergo molding multiple times as they have linear structure, and this makes them recyclable plastic. Whereas in thermosetting polymer, they cannot be molded due to the presence of highly cross-linked structure, and thus they are non-recyclable. Likewise, on the basis of chemical properties, plastic can be further classified into two groups, non-degradable and degradable. The term non-degradable describes polymers that do not break down to a natural, environmentally safe condition. Instead of its drawback, we prefer to use synthetic polymer like polyethylene, polyvinyl chloride, polypropylene, polystyrene in our day-to-day life, such as in lenses for glasses, grocery bags, etc. due to its flexibility, strength, ease of blending with other material and resistance to heat. In spite of its fringe benefits, due to the absence of efficient method for safe disposal of these synthetic polymers, they often end up accumulated in the environment, posing an ever-increasing ecological threat to flora and fauna. Polyethylene is believed to be carcinogenic and also causes bronchitis, skin disease, stiffness, and vision problems. Chlorinated plastics such as polyvinyl chloride and other categories of synthetic plastic produces methane, a highly powerful greenhouse gas. During the degradation process significantly causes global warming. This damaging outcome of synthetic plastic led scientists to explore whether plastic could be designed to become susceptible to microbial attack, making them degradable in a microbial active environment. Thus led to the evolution of degradable polymer. Now I pass over to Srija.
Thank you, Suparna. Now let's move to the degradation of synthetic polymers, as these are considered as environmental pollutants. Depending upon the degrading agents, synthetic polymer degradation, it can be classified into 4 types: photodegradation, thermal degradation, chemical degradation, biodegradation, where in photodegradation, light is an degrading agent such as UV light. In thermal degradation, heat is used as a degrading agent. In chemical degradation, chemical conditions affects the degradation processes, and in biodegradation, it is the microorganisms' biological activity. All these degradation mechanisms reduces the molecular weight of the polymers and allow them to get biodegraded. Thus, biodegradation is defined as physical or chemical change in synthetic as well as natural polymers caused by biological activity. Biodegradation processes vary greatly in synthetic as well as in natural polymers, as they can be degraded aerobically and anaerobically. Now, in aerobic biodegradation, aerobic microbes use oxygen as the electron acceptor and oxidize polymer into oligomers, dimers, monomers, and its byproducts are carbon dioxide and water. Whereas in anaerobic biodegradation, anaerobic microbes use nitrate, sulfate, manganese, manganese, iron, and carbon dioxide as their electron acceptor. And its byproducts are methane along with carbon dioxide and water. On the among all synthetic polymers like polyethylene, polypropylene, polystyrene, or polyvinyl chloride, low density polyethylene, a type of polyethylene, one of the major environmental pollutants, contributed by its hydrophobic carbon backbone and high molecular weight of the polymer. Hence, let's focus particularly on the biodegradation of the polyethylene. But polyethylene, in its natural form, it cannot be biodegraded. So to make them biodegradable, it requires modification of its crystallinity, molecular weight, and mechanical properties that are responsible for polyethylene resistance toward biodegradation. This biodegradation of polyethylene is known to occur by 2 mechanisms. First, hydrobiodegradation, where starch is used as an additive to blend the polyethylene, and this treatment makes the polyethylene hydrophilic, and thus consequently biodegradation gets catalyzed by amylase enzyme. Second, in oxobiodegradation, where transition metal acts as an additive and they catalyze the thermal peroxidation. In turn, microorganisms can easily access attack and remove this hydrophilic section and biodegrade low molecular weight oxidation products respectively. On the account of high molecular weight and high hydrophobic levels of the polymers, microorganisms are unable to transport them directly through the outer cell membrane into the cells. Thus, microbes, they developed a strategy to excrete extracellular enzymes such as lactases and manganese peroxidases capable of degrading polyethylene into low molecular weight fragments, and these low molecular diffuse into the cell and get assimilated in its internal environment. Subsequently, scientists started to explore whether plastics could be designed to become susceptible to microbial attack, making them degradable in a microbial active environment, and this led to the discovery of bio-based plastic, and its properties are biodegradability, apparent biocompatibility, and manufactured from renewable resources. Just like synthetic polymers, microorganisms can degrade Metabolize bio-based plastics. Here, enzymatic degradation by hydrolysis enzyme is a two-step process where monomers are being formed, and being water soluble, they passively diffuse to the cell wall to produce byproducts. Now, in conclusion, we can say that there are various plastic degrading methods available, but the eco-friendly one is degradation using microbes accompanied by blending with the additives. And because of the negative impact of the non-degradable polymer, the use of bio-based polymer, it reduces the environmental pollution. Thank you.
Yeah, it was a very nice presentation, and you covered all the basic aspects. So I really don't have, I couldn't find any like loopholes or anything in your presentation. So I have no question.
Thank you, sir.
Thank you, sir.
Very nice presentation. I also don't have any questions.
Thank you, ma'am.
Only one thing. Only one thing. Can I ask something?
Yes, ma'am.
Just the name of the enzymes. Just the name of the enzymes which are actually involved in the plastic degradation. Just name one.
Ma'am, in case of bio-based polymer, hydrolase enzyme was involved to produce the monomers. And in case of synthetic polymers, there was lactases and manganese peroxidases was involved. And these lactases and manganese peroxidases, they were of the fungal strain, and of the bacterial strain, like protease enzymes were involved.
Okay, very nice answer. Is there any mention about beta ketothiolase? Um, because in case of some of the natural so in case of some of the microorganisms which are able to secrete beta ketothiolase, they are also able to degrade these kind of complex polymer which are actually found in case of water contamination. So that's why when the Zuglial ramigera is used, so this Zuglial as Zuglial film, so this particular organism, they are also capable to secrete out this beta ketothiolase, by which they are able to degrade the complex polymer. Okay, but your answer is so good that you have mentioned about the hydrolases and the other enzymes, which are very much potent enzymes to degrade the synthetic polymers. Thank you so much. Very nice presentation. Very nice.
Thank you, ma'am.
Thank you, ma'am.
Thank you, Srija and Shuporno. The next and the last presentation by BSc second year, we have Piyash Mukherjee and Shirsho Roy.
Good evening, respected teachers, judges, and everyone present here. So I am Shirsho, and along with me is my partner Piyash, and today we are going to speak on the topic metagenomic sequencing with spike primer enrichment for viral diagnostics and genomic surveillance. So these are some of the objectives behind the study, but the main and sole thought process to carry out this study was to develop a novel bioprocess that would ease the process of pathogen identification. Now, at the outset, I would like to shed some light on what is metagenomics. So, metagenomics is a branch of genomics that deals with the study of genetic material recovered directly from the environmental samples. Then, metagenomic sequencing with spike primer enrichment. Well, what is that? So, it is just a target enrichment method. But why do we need such a method? So, to answer this question, I would like to go through the entire journey behind the development of this process. So, in the recent decades, we have faced several viral outbreaks, such as the Ebola virus outbreak, the SARS outbreak, and definitely the ongoing coronavirus pandemic. But the threats have been increasing over the past few years, and the possible reasons are: number one, rapid population growth, increasing urbanization; number two, and number three is increased global travel. Now, the control of the spread of the virus has always been a major concern in all these outbreaks. and the ordeals faced during the initial identification have always been either due to poor technological resources or due to the absence of diagnostic assays that could easily detect a new or a emerging strain. Well, MSSP was developed to serve this function. So metagenomic sequencing with spike primer enrichment, abbreviated as MSSPE, is a method for enriching the targeted RNA viral sequences while retaining the metagenomic sensitivity for the detection of other pathogens. It is used as an additional target enrichment step along with metagenomic next generation sequencing for broad spectrum pathogen identification and for creating the new and re-emerging viral strains. Well, we already know the available alternative methods such as the tiling multiplex PCR or the capture probe enrichment. But these processes have a lot of limitations, such as they are extremely expensive, they have a narrow scope of detection, they all follow complex and lengthy protocols, and nevertheless, they all have a big risk of cross-contamination. Well, all these limitations were taken into consideration and uh, successfully countered during the development of MSSPE. Now, in this study, MSSP was performed and evaluated for 14 different viruses, resulting in a 47% increase in the width of the genome coverage as compared to MNGS alone, while maintaining a 95% accuracy throughout the entire process. Now, as far as the discussion continues, MSSP has the following special features. It is a universally accepted robust target enrichment method. It follows simple protocols. It is relatively cheap and inexpensive, and it does not require any extra time between the cycles. It is not only compatible with the benchtop sequencers, but also compatible with the portable sequencers depending upon the available technology. Now, during the development of this process, a lot of other solutions did come up and they were tried upon the common pathogens, such as the host depletion methods. Now, these methods include complex phenomena such as differential lysis. So these processes were not only difficult to standardize, but were also found to bias the detection due to the increased amount of duplicated results. Now, this, as you can see, is the entire MSSP workflow. So the clinical sample is collected from the patients. This is followed by RNA extraction and DNA treatment. This is followed by reverse transcription with spike primers. Now, this is the most highlighted step in the entire process. Spike primers are named so because of their distinguishable appearance under the microscope and the fact that they are longer than the regular RH primers. Now, some random primers were also added in this step to aid the second strand synthesis. This is followed by the library preparation and the PCR amplification of the complementary DNA manufactured in the previous step. Now, all these steps were concluded by the final step, that is microbial identification using the SERPI technology, that is sequence-based ultra-rapid pathogen identification. Now, for the results part, I would like to pass the baton to Piyash.
Thank you, Shirsho. Now to begin with the next half of the presentation, that is the result, I'd like my friend Shirsho to take us to a slide that contains an animated flow chart actually elaborating the process of 13 nucleotide based spike primer construction that that Shirsho just explained a few moments earlier. Now, this slide actually deals, the rest of it deals with the clinical statistical data on the clinical performance of MSSP that will come across in the later half of the presentation. Shirsho, please. Uh, yeah, thank you. Okay, so here the first part of the presentation, as you can see, is an animated flowchart showing the construction of a 13 nucleotide-based spike primer for the identification of the viruses, the process that Shirsho just told. So when the scientists were preparing the primers, they actually prepared three kinds of primers. One is the virus-specific primer, the second type is the large primer or the large pool, and the third is the combined panel for all the 14 viruses that Shirsho mentioned over here. So as you can see in the data table given here, it is written ARBO-V. ARBO-V panel stands for arbovirus panel that will be utilized for detection of arboviruses like Zika virus, chikungunya virus, West Nile virus, dengue virus. So what's basically happening here, the number or the amount of primers the scientists had to invest per kilobase of the genome actually represents the genomic diversity of the species. So as you can see in the first row of the data given, Zika VSP, ZIKVSP actually represents Zika specific spike primer. And you see under the table, Shirsho is pointing with laser, the primers per kilobase of genome is 14.8. Whereas for dengue, in the last, you can see it's 54.5. So it basically means dengue virus, the genome is more diverse as compared to Zika virus, thing that the scientists came across while preparation of the primers. Please take us to the next slide, Shishu. Now, how does that matters? Because here we are talking about the primer enrichment methods that will be aiding processes like MNGS. So there, while utilizing a virus-specific primer, we need to know what's the concentration of the primer to be utilized and what is the amount of fold of enrichment we are getting. So as you can see in the table, in the last two column of the data given, ZIKV, that is Zika virus, for almost a concentration of 942 copies per ml of the filter, you see RH primer was utilized first, and then there was Zika specific virus, primer utilized at 4 μm concentration. And you see for that we had a 19 times, that is the 19 fold or 19 times enrichment of the Zika virus copies or took place in that contrived sample. And the graph that Shishu is now pointing with the help of the laser is the equivalent of the same experiment conducted on a larger panel, the arbovirus panel, the one I said in the last slide. So as you can see, the blue line actually representing the graph of enrichment of Zika virus in attains a maximum of 12 times enrichment along the y-axis when there is a 20 μm concentration of the SP primer utilized. And as you can see, the red line, interesting, in the last almost parallel with the dotted line, but not same with it. There's asterisk mark over it. It is actually the Ebola virus line. Sorry, the line of the Ebola virus enrichment. The Ebola virus here was the off-target species, but its metagenomic sensitivity was not hindered while utilizing the arbovirus panel or the Zika virus panel. Now, this is interesting. She should take us to the next slide. And this brings us actually to the merit of the clinical performance of MSSP. So while the scientists were pulling a CSF sample from a patient to utilize under arbovirus panel to find the re-emerging strains of HIV, they were able to successfully find, sequence, and increase the depth of coverage of PWV. In the green table, you see the last row, PWV, that is poison virus. It was also the off-target species, a re-emerging strain, and it was not their target, but it was still could have a 14.7 time of enrichment of the sample. Another question comes here. It is simply for viruses? No. It is also for DNA viruses, non-viral pathogen, and unknown viral contaminants. As you can see, both Klebsiella pneumoniae and HIV been enriched by arbovirus panel. You should take us to the next slide. So that brings us to the merit of this process as compared to other processes like tiling multiplex PCR or other processes. And the most important is here the amount of contamination is very low, as also the amount of duplicated reads generated. You see for MSSP, the duplicated reads generated mean is 20%, whereas for multiplex PCR is almost 80%. So here we can conclude that MSSPE, as is comparison to the other methods, is very efficient and economical method with almost no extra turnaround time, and most importantly, no contamination while pulling of the enriched sample, a tool that helps in real-time check for the efficient and effective viral genome coverage, increase in the depth of coverage, sequencing new or re-emerging viral genome, and also enrichment of different target viruses. That was the sole aim of this project of the scientists. Thank you. That was a presentation on metagenomic next generation sequencing for viral disease outbreak surveillance and genome coverage by Piyash Mukherjee and Shisha Roy.
Yeah, it was very interesting presenting a technique that is highly relevant, particularly as we are emerging into a world full of new pathogens.
Yes, sir.
So
Yes, sir.
it's a good choice. But I have one very basic question.
Yes, sir.
For metagenomics, usually people use degenerate primers. But here you are using spiked primers, which are a sort of a different variant of the common or regular degenerate primer. So my question is how spiked primers are different from regular degenerate primers? Why they are unique?
Yes, sir. The spike primers have a distinguishable appearance under the microscope, since some specific tags, the virus specific tags are attached to it, and also they are longer in length as comparison to the other degenerate primers, such as the RH primers.
No, that is not the correct answer. That is just some superficial difference
Yes, sir.
between degenerate primer and spike primer. This is not the major difference. Yeah, if it is tagged, you can also tag a regular degenerate primer, and you can visualize under microscope, or like electron microscope or whatever. And length, yeah, length can also vary. Even you can also vary the length of degenerate primers, but the still the actual answer is missing. You see, in degenerate when you make the regular degenerate primer,
Yes, sir.
you use equimolar mixture of ATGC, because you want to generate all the codons. You know, there are 61 codons, and there are many degenerate codons for different amino acids. So to cover all the codons, you use an equimolar mixture of ATGC.
Yes, sir.
But when you are designing a spiked primer, then you are reducing the degeneracy. You are not using the equimolar mixture. You are using non equal. So some of the bases are added in higher proportion than the rest, and you do not go for codon degeneracy. Now you are looking for codon bias. So different viruses and different cells, they prefer certain codons. So you are going for codon bias. So you get a higher specificity. So that is why they are called spiked primers. Some of the bases are added in higher molar proportions than the others. Okay?
Thank you,
Yes,
sir.
sir. Thank you,
Thank
sir.
you, sir. Thank you,
Yeah.
sir, for explaining.
Okay. So thank you. It was very nice to hear this.
Thank you, sir.
Thank you so much. Thank you. Very nice presentation, student. And thank you, Jaydeep sir. I don't have any. Thank you.
Thank you, ma'am. Thank you.
Thank you, ma'am.
So I believe we come to an end of the first technical session presentation by UG student, right? Am I right?
Yes, sir.
So I really congratulate all the students. Practically, everybody should know when the lockdown started in March 16th. That was March 16th, precisely, the lockdown started. And we were in totally in a troubled soup. We didn't know what to do. Then gradually we recovered. And I remember categorically we completed their presentation. I listened to all of them, shortlisted them, and I could find one basic difference. In the other years, when they are presenting in the stage, some stage fright is working. But here, speaking from home, they are speaking so spontaneously and with such confidence. It is really, I don't know, if I was the judge, it would have been very difficult for me, but they all presented so nicely. Congratulations.
Congratulations to you all. Very nice presentation. Okay, can I leave now?
Just one thing, you and JG sir, please uh, finalize the first three position and just communicate to the conveners, or whoever is the convener, you can communicate to him, or you can just give it to me also.
Okay.
So that the privacy is maintained. Thank you.
Yes. So I'm leaving Zoom meeting.
Thank you,
Thank
ma'am.
you.
Thank you
Thank
so
you.
much,
Thank you,
ma'am.
JD sir, for
Thank
your
you,
support and
sir,
your
and thank you all the
excellent,
students.
excellent suggestion, excellent question, excellent exposition. Everything was fantastic. And
Thank
that
you,
is what
sir.
modern trends is all about.
I'm mesmerized that all students, they are giving very nice presentation. Even my family members, they are also listening, and they are telling me, oh, it's wonderful how gracefully, how spontaneously they are delivering lecture. That can I add here. Okay, thank you so much.
অরূপদা,
বলো।
বলছি যে এই তাহলে এই আমার দেওয়া মার্কস আর এসএসসি ম্যাডামের দেওয়া মার্কস কি অ্যাড করে আপনাকে পাঠাবো?
অ্যাড করে তোমরা একদম ফাইনাল করে নেবে। আমার মার্কস পাঠিয়ে লাভ নেই, তোমরা ফার্স্ট
আচ্ছা।
3টে পজিশন ফাইনাল করে আমায় দিয়ে
ও
দেবে।
ও আচ্ছা আচ্ছা, তাহলে ওই 3টে দিয়ে দিলেই হবে। আচ্ছা
হ্যাঁ
ঠিক আছে, তাহলে আমাকে এখন তাহলে এসএসসি ম্যামের সঙ্গে যোগাযোগ করে ব্যাপারটা ঠিক করে নিতে হবে।
হ্যাঁ, ওইটা ফাইনাল করে আমাকে ওই নামগুলো তো তোমাদের দেওয়া আছে,
হ্যাঁ হ্যাঁ
ওইটার
হ্যাঁ,
একটা স্ক্রিনশট দিয়ে দেবে। ওই স্ক্রিনশট 1 2 3 লিখে স্ক্রিনশট পাঠিয়ে দেবে।
আচ্ছা আচ্ছা আচ্ছা, ঠিক আছে ঠিক আছে, ওকে
থ্যাংক
করে
ইউ।
দিচ্ছি, এখনই করে আপনাকে পাঠিয়ে দিচ্ছি। হ্যাঁ,
থ্যাংক
ওই
ইউ।
Thank you, sir.
হোয়াটসঅ্যাপে পাঠিয়ে দেবো। হ্যাঁ,
ওকে।
Thank you.
ওকে।
We shall now proceed to the third technical session, that is presentation by MSc third semester of our department. The judges for this session are Professor Dr. Onindita Banerjee and Professor Dr. Madhumita Maitra. The rules of the time limit remains the same, that is every group will be given 8 minutes and 2 minutes for question answer session. The first bell will ring at 8 minutes and the second bell will ring at 9 minutes, beyond which you will be interrupted by the judges. However, in case of technical glitches, you will get extra time. The first group presenting is Piyasha Banik and Ankit Kumar Nath.
Is the slide visible?
Yes, it is. Could you please go to the full screen mode?
Yes,
you can start.
Okay.
Yes.
So, good evening, everyone. Today, we'll be discussing about direct cardiac reprogramming from developmental biology to cardiac regeneration. I am Piyasha Banik, and let's get started. Every year, millions of people die worldwide due to a number of heart diseases. Both acute and chronic myocardial infarction is due to the tremendous loss of cardiomyocytes through apoptosis or necrosis. Human aging is also associated with an irreversible loss of cardiomyocytes. To compensate for the death of cardiomyocytes, scar tissue is formed by activated fibroblasts in the injured area. The human heart has limited regenerative potential, although postnatal vertebrates show low level of cardiomyocyte cell renewal. The endogenous regenerative capacity of the mice adult heart, although limited, comes from the differentiation of cardiac progenitor cells rather than replacement by existing cardiomyocytes through cell division. Cardiac progenitor cells are endogenous stem cells that contribute to the regeneration of cardiac tissue after injury. This lineage is able to efficiently transdifferentiate into several cell types like cardiomyocytes, endothelial cells, and smooth muscle cells. So, here the challenge lies in isolating such cardiac progenitor pools and to promote their expansion and differentiation potential in vivo. It has been seen that cardiomyocyte proliferation enhances in mouse models of myocardial infarction by overexpressing cyclin D2, periostin, fibroblast growth factor 1, and neuregulin 1. Recently, the addition of microRNA 199A and microRNA 590 has also shown to stimulate cardiomyocyte proliferation. Now, looking into another strategy to repair an injured heart, which is to supply new cardiomyocytes already differentiated from multipotent cardiovascular progenitor cells. This can form a promising source of cells due to advances in efficiency of human cardiomyocyte differentiation. So, a newly emerging strategy for cardiac regeneration is reprogramming of these non-myocytes in adult heart into functional cardiomyocytes. What is cellular reprogramming? It can be defined as the process of reverting mature specialized cells into induced pluripotent cells. For decades, we had the concept that once a cell terminally differentiated cannot be reverted back or differentiated again. But this notion was proved wrong with cloning of Dolly the sheep in 1997 by the technique of somatic cell nuclear transfer, which revealed that mammalian egg had ability in reprogramming an adult nucleus to the embryonic state. In the late 80s and 90s, Evidence suggested that cell-fate conversion can take place by a more direct route. Studies from the Weintraub lab demonstrated that fibroblasts could be converted into skeletal muscle cells in vitro with host expression of skeletal muscle master regulator gene MyoD containing homeobox domain. Thus began the race to identify such individual transcription factors that could guide cell fate in other lineages, including cardiomyocytes. Meanwhile, studies in model organisms resulted in expression of complex body structures with transcription factors having homeobox domain, and the best example is exemplified by induction of ectopic eye formation on the leg of Drosophila by overexpressing the eyeless gene. Thus, cell-fate conversion by bypassing developmental lineage and progression by altering the genes gave rise to some of the concepts in regenerative medicine.
So my name is Ankit Kumar Nath, and from this part I will take over. So we know about the Yamanaka factors discovered by Kazutoshi Takahashi and Shinya Yamanaka. So the factors were OCT4, SOX2, KEAP4, and CMYC. And these factors showed conversion of fibroblast into pluripotent stem cell, induced pluripotent stem cells, by the way. So this is something known as, now we are opting for something known as transdifferentiation. So what is transdifferentiation? It is basically conversion of one type of somatic cell into another type of somatic cell without conversion into intermediate pluripotent cell stages or progenitor stages. Next slide. So, with the advent of transdifferentiation, Zhu et al. Demonstrated the reprogramming of the exocrine pancreatic cells into insulin-producing endocrine cells were also possible using three factors. Those were NGN3, PDX, and MAFA. Now, this same kind of approach was also used by the scientist L. Huang, and he showed, along with his team, showed that GMT factors, or known as GMT cocktail, containing GETA4, MEF2C, and TBX5. So these all are transcriptional factors. Whatever I'm talking about is transcriptional factors, and these transcriptional factors are very much efficient in conversion of mutant fibroblast into myocytes. Now, what are fibroblasts? They are basically scar tissues, like she has already explained. So we have to convert this non-usable fibroblast into active cardiomyocytes. So this can be done with the help of GMT delivery, which is done by retroviral induction into the murine heart model. So Szabo et al. Along with this showed that along with the Yamanaka factors, if we inhibit the JAK-STAT mechanism, this can result in emergence of cardiomyocyte. Although the process, how it is done, is basically not found yet. Next slide, please. So how to experimentally induce the formation of cardiomyocyte? Basically, we take a transgenic mice which contains the alpha MY86 along with a downstream GFP tag. Now, what happens is that all cells contain the alpha MY86, the fibroblast and the non-myocyte and the myocytes. But the cells which are cardiomyocyte, they only express the alpha MY86, hence the GFP. Thus, whenever we are doing this kind of experiment using transgenic mice and we are inducing an artificial shock, myocardial infarction, all the fibroblasts that are converted, we can isolate them using the FACS. Because as they are fibroblasts, they will not express the GFP. So we can isolate them using FACS. Now we can do GMT, retroviral transduction, in vitro, and we can find that there is an emergence of stable reprogrammed cells. So positivity we can take home. Reprogrammed ICMs showed action potential like the adult ventricular cardiomyocyte. Along with that, they also showed transdifferentiation. But there was one difficulty encountered, that was the cells were reprogrammable, but they were quite low in number. So which is dealt in the next slide. Please, next slide. So when it was moved in vivo, the mouse, alive mouse, was taken and ischemic shock, or the artificial myocardial infarction, was done. And then we induce the GMT, not we actually, they actually induce a GMT cocktail with the help of retrovirus. And they found that non-myocytes were converted to induced cardiomyocytes within 4 weeks. And what kind of cardiomyocytes they were? They are all normal functioning cardiomyocytes with binucleated, the mature cells, they show binucleation structures along with normal sarcomeric repeats, and they generate action potential like the normal cardiomyocytes. And the transformation efficiency was much better than in vitro assays. The probable result might be because in vivo, the microenvironment is quite more suited for the emergence of the cardiomyocyte like cell lines. So result of this transformation can be further increased with the help of immunosuppressed mice, but this is not quite valid. It's just done experimentally, not for the original human trials. Next slide, please. So modification to the above process, which I already explained, can be done with the help of HAN2. What is HAN2? HAN2 is basically a transcription factor that could give rise to very highly stable cardiomyocytes, which shows a very much more resemblance to the endogenous cardiomyocyte with respect to the sarcomeric formation, the electrophysiological activities, and the linear studies have shown us that these cells were the resident cardiac fibroblasts, which are converted to the induced cardiomyocytes. Although there are different challenges we are facing, they are facing right now. That is, the first is low transformation efficiency. The transformation efficiency is very much less. Human trial needs to be done. Some cells show low electrical activity, although the diagram I've just shown in the slide over here is basically an induced cardiomyocyte, which is showing this kind of electrophysiological activity quite comparable to normal in vivo cardiomyocytes. And the challenge number 4 is FDA is very strong regarding any kind of viral based gene therapy, but there are some rays of hope, obviously. So recent MESP1 and ETS1 regulated conversion of myocytes to early cardiac progenitor cells are also observed. They are going on in trials right as of now. And lipoprotein lipase trials, which is going on in Europe using the viral vectors. So these also going, so it might be helpful for us to, for them to actually push the GMT cocktail into the this kind of trials as well. And next is it paves a way to develop treatments for diseases like liver diseases, for example, or different kind of kidney disorders, which can be cured with the help of this kind of artificial cocktails. But the transcription factors might be different than the original GMTs we are talking about over here. Next slide. So we like to conclude our whole presentation with this, that GMT cocktail is very much effective in conversion of non-myocytes into active cardiomyocytes. And this basically the flow like process which we are doing. We are taking a heart which is not working properly. We take retroviral induction and we then generate this kind of cardiomyocyte from the fibrotic scar tissue, which are the very result of the myocardial infarction. Thank you. The references for the studies are given in the slide itself. And in each slide, there is reference parts which I have mentioned for each kind of information we have gathered from those papers. Thank you a lot. We conclude our presentation with here. We are ready for questions.
Okay, Onkit, Priyansha, it was a nice topic you had chosen. First question from my side is, why did you choose this topic?
Ma'am, because these are newly emerging field with respect to microbiological aspect, that is the viruses. Okay, so like we are currently dealing with different kind of virus vectors like adenoviral vectors. We have baculoviral vectors along with retroviral vectors. So we've thought that induction of this virus for some useful purpose like remodeling of the damaged cardiomyocyte could be very much effective in treating different kind of diseases which are facing a lot of like medical drugs. For example, in case of liver regeneration, the drugs which they are giving, they can do two effects. One is they can actually induce more damage, and one is at times they're showing good effective positive sides of the treatment. So if we are doing this kind of processes where we are actually inducing the cardiomyocytes from non-myocytes in vivo only without any using any kind of drug, it might actually help. But although there are different testings and stages needed for this procedure as well. Thank you, ma'am.
And the second question is, since the paper has told that they are using retroviral transduction. Now, one of the side effects of retroviral transduction is mutagenesis, right?
Yes, ma'am. Yes, ma'am.
So, does this paper report something of that sort? This is one of the disadvantage of using retroviral gene therapy. So
Ma'am,
have they
as of now, yes, ma'am. As of now, they have not reported the mutagenesis part, but in vivo studies, there are some evidences which show that there can be like immunocompromisation of the thing. Like whenever they are trying to treat the mice, there must be some kind of reversion. The vectors which we are using, they are getting immunoneutralized from the process. So this can be a thing which is they are doing, and the mutation part. I think there might be some resources, but we have not encountered as of now.
So this work was published in which year? What was the date of publication of this paper?
This
The
was
paper.
around 2012 and another about 2006. The two mother papers we were working with.
Thank you, Ankit and Priyasha. I have no more
Thank you,
questions.
ma'am. Thank you, ma'am.
Thank you, Ankit and Pyasha. The next presentation is by Swarnika Roy and Suchandrima Bhaumik.
Good evening, everyone. Our topic for today's evening is biomimetic nanotechnology towards personalized vaccines. It is going to be presented by me, Swarnika Roy, and my partner, Suchandrima Bhaumik. So, when we first see this topic, two questions will arise in our mind. Why are the traditional disease management methods presently invalid, and what is the concept behind personalized medicine? Since the discovery of antibiotics in the early 20th century, the global expectancy of life has increased to 72 years of age. Antibiotics have revolutionized medicine by enabling treatments of diseases that would have been proven to be fatal ones. However, in the recent times, we have seen a downfall in the use of antibiotics due to a rise in the formation of superbugs. Superbugs are microbes with enhanced morbidity and mortality due to increased or high levels of antibiotic resistance. Similarly, the traditional cancer therapies have a history of ups and downs, not only due to the ineffectiveness of the treatment, but also the reality of complete remission and cure. As such, in the recent times, new therapies such as cancer immunotherapy are being introduced, and very recently, nanotechnology is being offered, which offers nanoparticles as alternative therapeutics. Becoming more precise and impactful in their approach towards the treatment of the patient under their care has always been the goal of all clinicians. This led to the development of personalized medicines, or what you call precision medicine. According to the Precision Medicine Initiative, it is defined as an emerging approach for disease treatment and prevention that takes into account the individual variability in genes, environment, lifestyle for each person. The basic difference between this and the traditional methods is the degree of reliance on data, especially genomic data, which is very important to make precise decisions about the treatments to be administered. Now, personalized medicine introduced to us the concept of vaccine nanotechnology. We know that the traditional or conventional vaccines have been very effective to us, but they have a lot of cons. Here is where vaccine nanotechnology is preferred because of the advantage, that is, they have the ability to co-deliver antigenic material along with the immunostimulatory adjuvants, which is very responsible for eliciting a greater immune response. Now, I would like Suchandrima to continue with the slides.
Now, coming to what is biomimetic nanotechnology, the term biomimetic stands for imitating the biological system. Biomimetic nanotechnology is a combination of nanotechnological techniques along with the normal biochemical processes of the cell to carry out targeted drug delivery. It is favored over conventional vaccination because it is highly specific. It utilizes only natural ligands, so there is very less toxicity involved, and it is effective against both cancer and bacterial diseases. Now, coming to the applications of biomimetic nanotechnology, the first one is targeted drug delivery. Nanoparticles, when coated with drugs and conjugated with lectins, bind to specific carbohydrate receptors of Helicobacter pylori, which causes stomach infection, and thus they deliver the drug to a very specific location. Second is the modulating activity of the biological targets. Viruses, when engineered, their properties are altered as a result of which it changes their ability to invade and infect host cells. The third is escaping the host immune system. Nanoparticles, when enters inside the host, are recognized as foreign, and the immune cells act upon it and can destroy it. Thus, they cannot carry out their function. This can be avoided by coating the nanoparticles with RBC membrane, which makes the immune system recognize them as self. Now, Swanika will continue.
Is the slide changing?
No.
Could you try once again, please?
Yeah. Yeah. So as Suchandrima mentioned, the term biomimetic refers to human-made processes, substances, devices, or systems that imitate nature. When this concept was combined with nanotechnology, it gave rise to a new era of treatment, that is nanovaccines. Nanovaccines have been used to fight against bacterial infections as well as cancer therapy. The first topic to be discussed today is antibacterial nanovaccines. Antibacterial nanovaccines are administered in many ways. One of the processes used are The use of membrane vesicle coated with outer membrane vesicle coated with nanoparticle cores. Outer membrane vesicles or OMVs are about 220 to 250 nanometer multi-antigenic nanostructures generated by the blebbing of the cell envelope of the bacteria. Their unique membrane protein profile is responsible for triggering high immune responses. Now, loading of OMVs inside a nanoparticle core, which acts, or a nanocarrier, which acts as an immunostimulatory adjuvant, is seen to trigger excessive immune responses or boost immune responses, as the OMVs on the nanoparticle surfaces are seen to more closely replicate the membrane protein profile during infection, thus providing better training cues to the immune system during the particular infection. As you can see in the figure in the slide, bacterial OMVs are taken along with nanoparticles, your gold nanoparticles, and these OMVs are coated with the gold nanoparticles. The gold nanoparticle, this unification stabilizes the gold nanoparticles and also aids in the multivalent display of OMV membrane antigen. As a result, a significant increase in the lymph node accumulation was also observed. Vaccination using OMV coated gold nanoparticles have seen to boost immunity to trigger immune responses by increased dendritic cell activation as well as E. coli binding IgG antibody production. Thus, this method of immunization is sought to be more effective against bacterial infection because it triggered both innate as well as adaptive immunity. Now, Shuchondema is going to continue.
So a definite treatment that can fully cure cancer is yet to be discovered because the pathogenesis of cancer is quite complex and cancer varies greatly from one patient to another. The traditional methods that are available to treat cancer often cause damage to the normal cells. So the use of biomimetic nanovaccine techniques provides us a better scope for treatment over the traditional methods. One of the processes used is non-specific immune modulation. Non-specific immune modulation involves the use of immunostimulatory agents that can recruit immune cells to the tumor site. Virus-like particles or VLPs have gained importance as immunostimulatory agents. VLPs are mostly plant-based and are devoid of nucleic acid. They contain the viral proteins that are assembled into capsid. And the process by which they trigger the immune system can be seen on the diagram on the left. Firstly, we see the viral DNA encoding capsid protein utilizes the plant translation and machinery to produce self-assembly of virus-like particles. These then migrate to the tumor and activate neutrophil and cause the secretion of chemokines. This is followed by tumor infiltration by the activated neutrophils and followed by tumor lysis and activation of T lymphocytes. VLPs have added advantage because they produce long-lasting immunity, and also being small in size, they can enter the lymphatic system, reach the lymphoid organs, and carry out effective antigen delivery and processing. VLPs are nowadays used in combination with chemotherapy to enhance tumor suppression. So in conclusion, we can say that biomimetic nanotechnology is proving to be effective against both bacterial infections and cancer. Since it utilizes the normal biological processes, it is being favored over other conventional techniques that involves the use of foreign substances inside the hostel, and being highly specific, it is a step towards the development of personalized medicine. Thank you.
Thank you, ma'am.
Okay, I have a simple question, or I just have a curiosity over the plant leaf which you showed, that it is acting as the, yes, for the production of implanter protein translation. Can you tell me what is basically happening?
Ma'am, this takes place mostly in cowpea mosaic virus.
Yes.
Like with respect to, so only the port protein, only the the nucleic acid is gotten rid of, and only the viral proteins are expressed in the plants.
Okay, so this means the viral proteins are coded by genes, and those genes are only expressed in this plant.
Yes, ma'am. And the virus, firstly, the virus is introduced into the plant. it takes advantage of the plant machinery to produce only the coat proteins that, and finally it forms the capsid, and then it is used as the VLP.
Okay. Okay. So it is devoid of nucleic acids.
Yes, ma'am.
Right? It is just containing the capsid.
Yes, ma'am.
And then the second question is, I couldn't understand that point, is why the immune system? You said that the immune system is less your evoked here in the case of biomimetic, right? You talked about immune system, I guess. So that point I couldn't understand.
I mean, this only is non-specific immune modulation.
Oh, no. Biomimetic, you're saying that it is imitating the biological system,
Yes,
the
ma'am.
nanoparticles. Yes, so it has got a preferred advantage, this kind of biomimetic technology. So I just wanted to ask that what are the advantages?
Ma'am, that I spoke about that. Firstly, it utilizes the biochemical processes. So there is not like no foreign substances are entering into the system. So no toxic byproducts are being produced. Firstly, that can damage other cells and other processes also. And secondly, it is delivering the antigen and adjuvant together. So it is like and it is delivering it to a very specific location.
Ma'am, can I say?
Yes, yes, definitely. Any
Yes,
one of you.
ma'am, biomimetic nanotechnology, which is being used in this process of immunization. And basically, what is happening since we are using nanoparticles and we are using the biomimetic process, which is actually the imitation of natural processes, biochemical processes. So when we are combining the nanoparticle size or nanoscale size, which very easily transports through the lymphatic vessels or all the other cells of the immune system, and when we are combining it with the biomimetic, that is the natural processes, it gives us a very advanced or a very high immune response, because there might be certain enzymes in the body which might degrade the toxic other suppose the other materials or substances that would have been otherwise introduced instead of these, like the normal immunization processes. So due to these two factors, that process is getting hindered, or it's preventing that process. That is why we are using this kind of nanotechnology or biomimetic nanotechnology.
Thank you, Shanika. Now I understood. Thank you. So I don't have any more questions if MM ma'am has.
Okay, ma'am. So will we stop sharing, ma'am?
Yes, you can stop sharing.
Okay, ma'am. Thank you.
Thank you, Swarnika and Suchandrima. The next presentation is by Madhura Mondol and Srijon Bhattacharya.
Yeah. So this is what our topic deals about, and during this lockdown, somehow or the other, all of us came to know that every one of us goes through a certain level of depression. And during this lockdown, this global pandemic, we got the opportunity to talk to one of the most eminent psychologists of Kolkata, Dr. Anuttama Banerjee, who inspired us by removing two basic myths that each one of us, or maybe maximum of us, had. That is, depression is abnormality. It's not. It's something very normal, and that is the first step to accept the fact that depression is normal, and yes, it is not abnormality. So, moving directly onto the topic, epigenetic, the dark secret behind depression. Today, my partner Modhura and myself, Srijan, will be talking on this. Directly going to the statistics given by the World Health Organization, we can see that depression is a common illness worldwide, which can be compared with diseases like cancer, like diabetes, and many others, where more than 264 million people are affected with depression. And almost 8 lakhs people die due to suicide every year. And suicide is the second leading cause of death in 15 to 29 years old. Now, the saddest part is the most common treatment are the therapies. There are lots of kinds of therapies and the pills or the antidepressant pills like SSRI, etc. But these pills have lots and lots of side effects. And these therapies in countries like developing countries like India and countries with low income group and middle income group, 75% to 80% of the population do not have the ability to get that kind of treatment. Now, one of the particular things which we miss very often is that maternal mental health, and yes, we know how much the woman health is not considered in the country like we live in and globally. So, moving on directly to our topic, epigenetic inheritance of depression. To know about epigenetics, first we have to understand what's that. It's a modification of the gene expression rather than altering the whole DNA or the genetic code itself. There are lots of mechanisms in epigenetics like methylation, acetylation, histone modification, and etc. that we'll be talking about in the later part. And there are already many proofs where it is clearly said that epigenetics is the leading cause for the inheritance of diseases. Like here is a small example: a woman who smokes, maybe passively or actively, have the effect in up till its third generation. Now, moving to the next part, that is the maternal inheritance and accumulation from early life. We will majorly focus on the maternal inheritance part. Talking about the maternal inheritance, there are several parameters which are considered to have epigenetic modifications. We have searched for the literature, we have done the literature review, many hypotheses, many claims where it says that maternal stress, mental stress, the malnutrition, the mistreatment during pregnancy, or any kind of infection during pregnancy can lead to epigenetic modifications. And talking about environmental factors like bullying, like comparing, something that can create a mental spot or a mental darkness inside us. This all have epigenetic modifications, and the results are underweight, which is already proved, obesity, that is another thing proved, asthma, major health problems like cancer, diabetes, etc. And then coming to depression, many hypotheses have been given, but it is yet to treat it, or the research is yet to done in the basic level. Now going to the first part that is already being talked about, the HPA axis, where the after getting a stress, the hypothalamus gives out CRH, which directly activates the pituitary gland to give out ACTH, and finally the adrenal glands give out cortisol. Now this cortisol gives a negative feedback to the hypothalamus. But for getting that negative feedback, the glucocorticoid receptor have to accept the response. But this receptor is being controlled by the gene NR3C1, which is very much prone to epigenetic modifications.
Now that we have got an idea about how the epigenetic modifications control depression, we'll dive deep into the molecular mechanisms that guide the expression of genes related to depression. Now, there's a protein called the glial cell line derived neurotrophic factor, or the GDNF protein, which is expressed mainly in the nucleus accumbens region of the human brain. And it helps in the differentiation of the dopaminergic neurons, which is directly related to the release of the happy hormone dopamine. Now, in case of a chronic stress, the histone deacetylases are activated, which leads to the deacetylation of the histone, which in turn causes chromatin compaction. One more effect is that in the particular CPG sites in the promoter region of the GDNF gene gets methylated, after which a protein called methyl CPG binding protein recognizes this 5-terminal methylated cytosines, and along with cyclic AMP response element binding protein, it binds onto the promoter, turning off the GDNF gene, as a result of which the production of GDNF decreases and the dopaminergic neurons are suppressed and the production of dopamine is halted, triggering depression. Now, how is this epigenetic modifications and its heritability, if it can be at all predicted using stochastic modeling? So in this case, we took a particular case of methylation of a lysine residue at the 9th position of histone H3. Now there's a protein called heterochromatin protein 1, or HP1, that activates the methyl transferases and
Upon methylation of the histone, it will bind to those methylated histones, after which it will oligomerize and bring the adjacent nucleosomes into close proximity, leading to compaction of the DNA or the change of DNA from the euchromatin to the heterochromatin state, as it is evident in the picture on the left-hand side. Now, this particular mathematical model is based on Monte Carlo simulations, which is widely used in biological systems because it takes into account the intervention of random variables and it then predicts the probability of different outcomes. This has taken into consideration the thermodynamic stability and configurational statistics of the methylation system and a kinetic equation for the change in methylation probability with respect to time was found out. So we see that this rate of change is dependent on the methyltransferase activity, the probability that a particular nucleosome remains unmethylated denoted by 1 minus pi, and as well as the demethylation rates. Now, whatever was predicted using the mathematical model, it was cross-verified and supported by experimental data. So here are the results. This shows the representative configuration of the chromatin structure at 3 generations, that is 0, 2, and 4, for different concentrations of the heterochromatin protein. So we see that a very high concentration of the heterochromatin protein, the methylation is gradually increasing across the generations as denoted by the red lines in the graph. And at a very low concentration of the heterochromatin protein, the methylation is gradually lost across the generations. But for an optimal concentration of the heterochromatin protein, that is at 0.7 micromolar, we see a steady state of methylation being maintained across the generations. Now, this particular slide focuses on that whether the compaction state of the chromosome is at all dependent on the presence and the number of the neighboring nucleosomes which are bound to HP1. And as we can see very clearly, that those zones which are denoted by purple are the heterochromatin nucleosomes, and from the graph on the right-hand side, we can see that the number of neighbors associated with HP1 is highest for heterochromatin and least for the unmethylated or euchromatin nucleosomes.
Now, what actually we tried doing, we did a survey based on the Beck's depression inventory, where we tried to gather the fact about the depression, the levels of depression in the individuals and their mothers, and the environmental factors. We used to collaborate between the qualitative as well as the quantitative biostatistical data that we received by using the tool, namely RStudio. And there are a few very random yet very important responses which will really shock us. And these are just the glimpses.
Now, moving on to the final quantitative correlation of maternal inheritance of depression. The depression indexes of 148 individuals and their mothers were correlated, and it was graphically represented. And we carried out a correlation test using RStudio, and we found out the coefficient. It turned out to be 0.36, and it lay in the 95% confidence interval. And this value of 0.36 clearly denotes a positive correlation between the depression index of the mother and her child. So we can, based on our survey, we can actually kind of predict that yes, the depression of the mother actually affects the depressive state of the child.
And there are few limitations of our topic, like of our thing that we did, the research kind of study we did, that we focus on a very small group. We couldn't do a face-to-face interaction, and the evaluation of the depression process needs some more factors to be considered. And the loci, the future prospects, where we should focus on the loci of the epigenetic modifications, and this understanding of the loci will ultimately help to epigenetically fine-tune the diseases which are global concern. And if we can do that fine-tuning in the stem cell level, it can focus on the embryo level treatment in future. Now, these are the very few references out of many we have used. And finally, we would like to thank you, each and every one of you present here and listening to us. Thank you.
Thank you.
Okay, question for Srijan. Can you tell me what is the
biometric pattern of the measurement of depression?
Yes, ma'am. Can you please repeat the question? Ma'am, can you please repeat the question?
The biometric pattern for the measurement of depression.
Ma'am, in this process, the Beck's depression inventory or the biometric process, there are lots of biometric process actually, but we use the Beck's inventory where we had 21 questions, and on the basis of the 21 questions, according to the responses are getting marked from the 0 to 3 pattern, and after summing up the total calculation we get, we put that value into certain ranges, and that range tell whether the person is just anxious or mild stress or moderate depression or extreme depression, like this.
And how does this pattern is dependent on the methylation? I mean, how do you correlate with methylation?
Ma'am, actually, through the process of depression, why what we try to see that whether the depression range of the mother and the depression range of the child is coinciding or not. That we tried to show in the graph that Moduna talked about, where she showed that the depression pattern is almost similar for the maximum cases. And then from there, there have been lots of proves that epigenetics is this cause for the inheritance of major diseases, and that same goes for the depression pattern.
Okay, okay. Thank you, Srijan.
Thank you, ma'am.
Srijan and Modura, very interesting topic. So you did this survey, I guess. You sent me a questionnaire too. I had forgotten to fill it up. Now I understand why. I had forgotten to fill it up. With so much
Yeah.
of class load, I had just forgotten. But now I could make out
Actually,
why you
it's a
had
very much
sent it to me.
anonymous process, yeah.
No,
Okay, it's
nicely
very
presented
interesting.
and nicely done. A very
Yeah, very
good
nicely
topic
done.
to be selected, especially during this hour.
Yes, we all are little bit depressed because of the environment surrounding us. So that's true, but I never thought that epigenetics can have a role in this. That if we become depressed, why we are becoming depressed, also there is an answer to it, right? So I never thought it in this way, that epigenetics again has a role to play here. So have they studied any other kind of epigenetic marker beside what you talked about, beside you talked about the methylation of the CPG sequences at the promoter? So beside that, is there any other report?
Besides methylation, we have also studied about deacetylation of the histone proteins. What happens in case of deacetylation is that once a stress is induced, the histone deacetylases are activated, and it causes deacetylation of the histone proteins mainly at the H3 subunit. After deacetylation, the DNA can more tightly wrap around the histone octamer, resulting in a compaction of the chromosome. So what happens is that region of the chromosome does not remain accessible to the transcription factors for transcription and then the expression of those genes. Uh, for example, genes coding for hormones like dopamine, the happy hormones like endorphins, oxytocin, all those things. That is how the stress is related to the expression of certain hormones.
Okay, so that's dopamine and cortisol. So whenever we are stressed, this means that the central nervous system, it releases your, like you showed us at the figure, and finally it ends up in releasing cortisol, right? During stress, so cortisol is released.
Yes,
How
ma'am.
does cortisol act? Why do we, under stress, cortisol is released? Okay, we know that. It's a stress hormone. So then after that, how does it affect
Ma'am, actually,
cortisol?
it serves as a negative feedback to the brain or the hypothalamus. Now,
Yes.
what happens? Yeah, what happens that there are glucocorticoid receptors in the hypothalamus, which on getting this stimulus, it stops producing more CRH, corticotropin releasing hormone. So once it stops producing that, now the stress, it's no more CRH is produced, and the action that we do due to the stress is now stopped. So, but if the GR parts like the glucocorticoid receptor, the genes encoding that, like the NR3C1, as we talked about, if these genes, which are very much prone to epigenetics modifications like the DNA methylation, then what happens? Then they cannot receive that kind of negative feedback. And then the hypothalamus keeps on producing the CRH, and the whole process goes on, and this trace remains. And finally, what happens? This leads to hypermethylation of that part. We know that the part which is already methylated tends to get hypermethylated. So this leads to the inheritance of the whole process, and that stays or that persists for the next generations.
Oh, very well explained, Srijan. Thank you. Thank you.
Thank you, ma'am.
Okay. Should I stop sharing now?
Yes, you can stop.
Okay, thank you.
Thank you.
Thank you so much, Madhura and Srijan. The next presentation is by Shobhik Banerjee and Ankita Datta Chaudhary.
Good evening to the respective judges and all the participants and spectators of Modern Trends in Microbiology, Chapter 17. Today, I, Ankita Datta Chaudhary, and my friend Shobhik Banerjee, we are going to present a paper titled Physiological Amyloids, an approach towards decoding the mystery of neurodegenerative diseases. Now, before going inside the topic, there are three terms which need to be very clear to us. First, what are amyloids? Amyloids are nothing but, in simple terms, aggregation of proteins. What kind of proteins? Beta sheet structure of proteins. So, this kind of aggregation are found in two types. First, the pathological amyloids. Now, we all know very well what are the pathological amyloids. They're very common to us. This kind of amyloids are found in the brain, and the aggregation of proteins, the insoluble aggregation of the beta proteins in the brain, they basically cause different types of diseases like the Alzheimer's, the Parkinson's, and many others. But now we are going to study about something called the physiological amyloids, which are not very common to us. The physiological amyloids are a type of aggregation of proteins of the beta sheet structures, which are found in the early oocytes of many vertebrates, including the mammals. So let us study and go a bit deeper into the physiological amyloids. Next slide, please. So, going into the physiological amyloids, we first need to know what are the Balbiani bodies. The Balbiani bodies are nothing but non-membrane bound compartments or organelles which are very rich in mitochondria, RNA, Golgi, and many other protein aggregations. These Balbiani bodies are the main components of the physiological amyloids, and they are responsible for protecting the early oocytes. Now, analysis of the xenopus oocytes and orthologous buckyball oocytes in the zebrafish have shown very clearly how the Balbiani body looks like. We can see the very dark spots are the mitochondria, the green marked one are the RNA particles, and the yellow marked part are the Golgi bodies. Next slide, please. Now, going a bit deeper into the Balbiani body, analysis of the Balbiani body shows that there is a mystery protein, the X-velo or the velo-1 protein. Now, why this is known as the mystery protein? Because this protein is a highly disordered protein and no conserved domain has been found in this velo protein. But it has been found that the Balbiani body is very much enriched in this protein, and the Balbiani body is linked to the X-velo protein through the C-terminus with positively charged amino acid residues. Linking of the Balbiani body with the X-velo protein is very important in protecting the oocytes in the early stages. Now, as I said, that the X-velo is a very disordered protein and no conserved domains have been found in the X-velo structure. But one conserved domain has been analyzed. And what is that? The prion-like domain, which is mainly responsible and is present in the N-terminus of the X-velo protein. The prion-like domain is the most conserved and rather the only conserved structure of the X-velo protein, and it is mainly rich in aromatic amino acid residues. It has also been found that if the prion-like domain, the amino acids in the prion-like domain is substituted with positively or negatively charged, the prion-like domain completely disrupts. So as far we have known till now that the prion-like domain is the only conserved in ex vivo, and the ex vivo is linked to the Balbiani body, which protects the early oocytes. Next, please. Now, you may ask now, you may be thinking that why we were talking about amyloids till now. This ex vivo network, that is stimulation of the prion-like domain in the ex vivo network, leads to the formation of the amyloid-like structures. The prion-like domain, when it is mutated, completely leads to the disassembly of the ex vivo network, and this is how these physiological amyloids get degraded. So basically what we have seen from the analysis that when the early oocyte develops into a mature oocyte, the prion-like domain somehow gets dispersed and the ex vivo protein is completely disrupted, which leads to the disruption of the Balbiani body. Now, we tried to draw a link between the physiological and the pathological amyloids. We have seen that the physiological amyloids can be degraded very well. But in case of pathological amyloids, it has been known to us that the pathological amyloids cannot be degraded easily. Now, but we have seen that both the physiological and the pathological amyloids consist of the prion-like domain structures. So basically what we wanted to do is we perform some experiments of the very little knowledge of bioinformatics and computational biology that we have and tried to draw a possible relation between them and try to draw a future scope of how using the pathways of the physiological amyloids and analysis of the different ex vivo proteins, the pathological amyloids can be degraded. I pass to the next part of the presentation to my colleague Shobhik Banerjee. Shobhik, please take over.
Thank you, Ankita. So in this presentation, instead of just reviewing a paper, I will present certain data that we got from various computational analysis which we have performed on our own. So first of all, you can see the secondary structure of the veloprotein, which has been predicted by the Cypred software. On the left-hand side figure, you can see that the Cypred analysis shows that the protein has majority, that is 77% of its structure as coil structure. Like that means it is, Ankita has already said that this veloprotein is a disordered protein. So from this particular result, it is very clear that this particular protein is a disordered one. Now, I just want to tell one thing, that sequencing of this protein has already been done, but its structure is not yet determined. So we just tried to build a model of this protein using the tertiary structure prediction tool Fire Square by using the concept of homology modeling. So as Ankita was telling earlier that Velo is a disordered protein, we just tried to validate the statement using the Ponder software. In the graph, you can see the red peaks, and those red peaks are actually indicating the disordered sequences. Among 779 amino acids of this Velo protein, 343, that is almost 44%, were shown to be disordered. And it has already been discussed that the veloprotein has a prion-like domain and it can form amyloid aggregation. So we used Plaque and Prion W, two tools, to check the presence of the prion-like domain. In the figure below, you can see that 60 amino acid long sequences is actually the prion-like domain, and the 21 amino acid residues marked in red is the amyloid core, which is responsible for this aggregation. Now I will come to a very interesting point, that during the maturation stage of xenopus oocytes, the amyloid aggregation is degraded. So there must be a degradation pathway taking place in case of physiological amyloids. From pathway analysis by string database, we can see that the xenopus velo interacts with another protein called ZFAN-D4, which has a ubiquitin domain. The ZFAN-D4, you can see from the pink box, the full form is AN1 type zinc finger protein 4, which has a ubiquitin domain. Interestingly, we found, you can see the right hand side picture, that this ubiquitin protein also interacts with PSMB2 protein, which is nothing but a member of the proteasomal complex. So it is very clear from these pictures that physiological amyloids are getting degraded by the ZFAN-D4 PSMB2 pathway, or simply the ubiquitin proteasome mediated degradation pathway. So an obvious question will come to our mind, that whether the ZFAN-D4 ubiquitin protein is present in humans or not. To find that out, we just checked the expression profile of the protein in human brain from the EMBL EBI expression atlas, and you can see that it is getting expressed even in the amygdala cells, which is affected badly in case of Alzheimer's disease. Next, from the cluster omega analysis also, we found that xenopass and the human Z-FAN-D4 proteins, they share 81% sequence similarity in their ubiquitin domain. And this is a very interesting observation according to us. So why the pathological amyloids are not getting degraded by the same mechanism? We also checked the human Z-FAN-D4 pathway and found that the protein can interact with a number of proteasome complexes, as you can see, PSMA1, A2, and so on. But to our surprise, we found that unlike xenopass Z-FAN-D4, the human ubiquitin protein is also interacting with two protease inhibitors, A2ML1 and SERPIN-B4. So physiological amyloids can degrade by this ubiquitination proteasome mediated degradation mechanism, but pathological amyloids are unable to do that just because of the presence of those protease inhibitors. Drug designing can be against these two protease inhibitors can be an effective therapy against neurodegenerative diseases, as and we have also uh, and we have also started to virtually screen a number of small molecule ligands from FDA approved drugs and the drugs which are in clinical trials. We have also started the docking process of those ligands using Autodoc Vina, and we are still waiting for a good result. Hopefully we will get something in future. So these are the summaries and the future scope slides. I have already gone through everything, the veloprotein, physiological amyloids, protease inhibitors, and obviously the future scope of drug designing. And finally, these are the references. These two papers we have referred to, one from Cell and another from Cell Cycle, a journal of Taylor and Francis group. And I just want to mention that both of these papers are authored by Elvan Boak, and she is currently working at the Center for Genomic Regulation, Barcelona, and she did most of her work at Harvard Medical School during the postdoctoral studies. So thank you for your patient hearing, and if you have any question, you can ask us.
Okay, Shobhik, I have one question to you.
Yes, ma'am.
Can you tell me a very prominent difference which is there between the pathological amyloid protein and a non-pathological amyloid protein?
Yes, ma'am.
Can you tell me the difference, the main prominent difference by which you can make it out?
The pathological amyloid and the physiological amyloid, the prominent
Yes.
difference.
Yes. How do you make it out? Because you have to target it. So how do you make it out?
Yeah, ma'am, actually, we are not targeting the amyloid proteins, but we are targeting the pathways, the cell signaling pathways by which they are getting degraded. The physiological amyloids are getting degraded by the ubiquitination proteasome pathway,
Yes.
but
Yes.
pathological amyloids are not getting degraded because they.
Yes. So you have to.
Hmm.
So any
Yes.
idea why that is the reason where which lays behind that why the pathological are not degraded?
Actually, we can see that the pathological amyloids are not getting degraded because of those presence of those protease inhibitors. Those protease inhibitors are actually inhibiting the role of the proteasome complexes. So the degradation mechanism are not working properly in case of pathological amyloids, but the physiologicals are getting degraded because there is no presence of those protease inhibitors.
And about the role of mitochondria?
Okay, ma'am, I'm not sure about the role of mitochondria in this part.
Okay, okay. Well presented, Shobhik and
Thank you, ma'am.
Anita.
Thank you, ma'am. So can I stop sharing the screen?
Well
Yes, you
presented.
can.
Okay. Thank you, ma'am.
Ankita and Shobik, very well presented. And I really like the bioinformatics part. So SSC ma'am is not there. I guess if she would have been there, she would have been very happy because she teaches you all of this. It's very nice presentation. I have no question. Thank you.
Thank you so much, Shobik and Ankita. The next presentation is by Samarpita Ghosh and Tanni Nag.
Good evening, everyone. Today, I, Ponni, and Shamapita are going to present the topic a novel Syndrome virus DNA vaccine and its inclusion in a candidate ban hantavirus against hantavirus pulmonary syndrome, HPS, and hemorrhagic fever with renal syndrome, HFRS. So, moving on to the introduction about hantavirus, hantaviruses are a family of viruses which was discovered in Asia during Korean War, and it belongs to the family of Hantaviridae in the order of Vanya virus. It is a zoonotic virus which causes infection in rodents but do not cause disease in them. It causes diseases in humans when they come in contact with rodent feces and urine. Next slide. So this slide presents the classification of the Banyaviridae family, which consists of 5 genus, and among them, we are mainly focusing in this presentation on the hantavirus genus, which contains 4 main types of viruses, which are Humala virus, Synombe virus, Andes virus, and Hansen virus. And the next slide will be told by Shomolpita. Shomolpita.
Hello, am I audible?
Yes.
Yes, you are.
Okay, I will be talking about hantavirus genome. Hantavirus is a RNA virus. It is a single-stranded RNA virus consisting of 3 segments of RNA. It is consisting of mainly the tripartite genome, and the 3 portions of the RNA segments are named as S, L, and M. S encodes for the nucleoprotein, and the L encodes for the RNA-dependent RNA polymerase, whereas the M encodes for the glycoprotein, surface glycoproteins of the virus. So we are coming to the pleiotropic function of the N glycoprotein. The term pleiotropic is generally is related with the fact that a single protein might have multiple functions. So this N glycoprotein or N nucleoprotein has a crucial role in cap snatching. So any kind of negative sense RNA virus has these kinds of activity, and these cap snatching mechanisms are also being observed in influenza virus. And the N glycoprotein can also functionally replace the eukaryotic initiation factor 4F cap binding complex. So it prevents the translation machinery of the host cell. And these N glycoproteins are also encoding a particular segment which is required for the RNA binding, or this is known as the RNA binding cleft. Now, facts on N glycoprotein. We have addressed a kind of protein which is also known as the tumor suppressor protein, that is PDCD4, and it has 2 domains, MA3 domains. These PDCD4 were interfering with the activity of eukaryotic initiation factor 4A, which is the ATP-dependent helicase in case of the eukaryotes, and was stopping the translation machinery of the host cell. Now, similar kind of activity was observed in case of the N glycoprotein. So these N glycoprotein was shown to substitute the entire eukaryotic initiation factor function, and it also promoted the preferential translation of the viral RNA. So coming to the host range, we are generally concerned with the 2 types of host, primary host and secondary host. Primary host is the ultimate host, that is the human beings, and the secondary hosts are in this case, are the deer mouse. So now coming to the infection cycle. Infection cycle, generally, this virus, generally the infection cycle takes place in the endothelial cells, epithelial cells, macrophages, and follicular dendritic cells. Mainly, we are concerned here about the hantavirus pulmonary syndrome. So these cells are being infected by the virus in the lung. So the attachment occurs in case of the glycoprotein present on the virus cell surface with that of the beta-3 integrin present on the cellular surface. Now, there is a gradual endocytosis followed by a fusion of the endosome membrane with that of the viral membrane and release of the viral particles into the cytosol, followed by the translation and transcriptional machinery and the replication machinery for the multiplication of the virus and release of further more virus into the host body. Now, we are concerned with there are two types of hantavirus which are generally present, that is old world hantavirus and new world hantavirus. Old world hantavirus generally causes HFRS syndrome, and it is mainly prevalent in Asia and Asian region. And new world hantavirus, which we are concerned about in this presentation, generally causes hantavirus pulmonary syndrome. Hantavirus pulmonary syndrome is much more severe than HFRS syndrome because it is having much higher mortality rate, and till now there is no such vaccines for the virus. Over to Tanni.
So, getting into the details about hantavirus pulmonary syndrome and hemorrhagic fever with renal syndrome, HPS is a serious fatal and zoonotic disease which is caused by the new world hantaviruses, namely Cinomdevirus and Andis virus, and they cause symptoms like mild fever and headache, which can lead to serious symptoms like shortness of breath, cardiac shock, leading to respiratory failure. No antiviral therapy is available till now. And HFRS, next slide, Samosita. HFRS is another serious disease which is also called Codian hemorrhagic fever, caused by the old world hantaviruses, namely Pumalavirus and Hansen virus. And the incubation period is about 3 weeks, and the symptoms vary from fever to proteinuria. And the treatment includes ribavirin, intravenous ribavirin, which is used to decrease illness associated with HFRS. Next slide. So the fatality rate of HPS is about 40%. Individuals with HLA-B5301 has got high risk of developing severe HPS because they show high levels of CD8 plus T cell response to SND. Next slide. So when the lung tissues from HSC patients were diagnosed, high levels of CD8 plus T cells and cytokine producing cells like the tumor necrosis factor alpha and interferon gamma were observed, which might have led to the alveolar capillary leakage inflammation leading to respiratory failure. Next slide. In immunohistochemical staining techniques are used to enumerate the cytokine producing cells. Next slide. Now, there are different factors that play a role in the entry and infection by the two types of hantaviruses, and ITAM. PCG1 is category 1, which is an essential factor that helps to virus to enter and bind and infect the pulmonary endothelial cells. And ITAMs are also the main signaling elements which are found in and dysregulate normal and immune and endothelial cell responses and send extracellular signals to intracellular signaling pathway and activate two kinds of kinase family like SRT and SYK family. Now, here in this experiment, we prepared the SND vaccine. For preparing the vaccine, strains were propagated in vedo-eated cells and maintained in eagle's minimal essential medium. And the model organisms used here are the female New Zealand white rabbits and female Thailand hamsters. And the next slide will be taken by Shomavita.
Now, we know that hantavirus are the RNA viruses. So we need to reverse transcribe the M gene. M gene is particularly immunogenic to the host cell. So we need to take up the M gene and reverse transcribe the whole gene. And followed by the PCR amplification, we need to prepare the cDNAs. Followed by, we need to inject those cDNAs into that of the plasmid. And we generally prepare a DNA vaccine which is named as PWRGSNM2A. Followed by, there was also a preparation of second kind of vaccine where the M gene was being optimized and further injected into the host cell. Now, why do we use DNA vaccine? DNA vaccine is mainly used because it can kind of activate both the cell-mediated immunity and the humoral immunity. So here we mainly use the muscle electroporation vaccination to inject the rabbits with the prepared plasmid DNA. So what is plate reduction neutralization test? Plate reduction neutralization test is used to quantify the amount of antibody that is required to neutralize a particular virus. Now coming to the results, we are going to individually inject the plasmid DNAs which are we are going to inject the plasmid DNAs into the rabbits, and we generally observe the result. We see whenever we put the M gene segment vaccine, that is vaccine 2A, was immunogenic in rabbits, but after a few days it failed to give considerable results compared to when we provided the rabbits with injected the rabbits with optimized version of the vaccine. It gave a good result and a good titer of antibodies. Over to Tanni.
Now, directly moving on to the results of the next section, here we made bivalent and multivalent vaccines. Next slide, Chamatita. And the results showed that there are increasing level of antibody titers against the SNV and ANGV in the HPS vaccine and multivalent vaccines, and high levels of increased levels of antibody titers were found against PUV and HPNV in the HFRS and multivalent vaccines also. So this suggests that it is possible to mix the hantavirus DNA vaccine into a single injection vaccine and produce neutralizing antibodies against multiple hantaviruses. Next slide. To determine the protective efficacy of our SNV vaccine, hamsters were vaccinated by gene gun method, and the results showed high amount of antibody titers, which could protect the hamsters only after 3 vaccination processes. Now, we also made an experiment to find out if our SNV vaccine is protective against ANDV. Next slide, Shamatita. Results showed that only 3 out of 8 hamsters survived despite producing high levels of antibody against SNV, which clearly showed that it failed to provide protection against ANDV. Next slide. So now we conclude by saying that very successful SNV DNA vaccine was produced, which could provide protection to both the animal models by muscle electroporation and gene gun method respectively. However, cross-neutralization was also found against all the 4 hantaviruses in the which vary depending on the route of delivery. Thank you.
Samarpita, can you please stop the screen sharing?
Yes.
Ma'am, do you have any questions for this team?
Hello, can you hear me?
Yes, ma'am. Yes, ma'am.
Okay, I don't have any as such questions, but I just have one or few suggestions to Tanni and Samarpita. Please don't write so much on your slides. Okay. So you have written a lot on your slides. If you write so many things, it becomes very difficult to follow. Either you give us time to read or you explain it in a very proper way, in a nice. Otherwise, I don't have any questions.
Okay.
Thank you, Samarpita and Tanni. The 6th and the last presentation of today is by Prapti Rosario and Meenakshi Garg.
Can we start?
Yes, you can.
Okay. Okay, so a very good evening to all the judges and the spectators present with us today. So today, me, Prapti Rosario, and my teammate, that is Meenakshi Garg, we would be presenting a very intriguing topic this evening. It is that a highly parallel lab evolution reveals that epistasis can curb the evolution of antibiotic resistance. Okay, moving on. So it is no secret that bacterial resistance to antibiotics is happening, and it is a major concern of public health and also a huge field of research. So this benefit is only limited to the bacterial world, not to us. For us, it comes with our own set of problems and a lot of research. So to counter this spread of resistance, there are certain steps which have been taken. To name a few, it would be discovery of new antibiotics involving different drug combinations, novel treatment schemes, and also efforts are being made to identify genetic factors or cellular mechanisms which not only stop or hinder the pathogen's resistance mechanism, but also slow down its evolution process. And lastly, epistasis. Epistasis is what our paper is based on, so it will be discussed now by my teammate.
So now exactly what is epistasis? Epistasis is a gene interaction where one gene masks over the phenotypic effect of the other gene. So in here, we have to see that it's not homologous alleles, but rather it's two different genes. In this study, we study two different types of gene mutation. One is the spontaneous mutation, which is determined by the genetic background of the cell. And the other is perturbation of cellular functions, such as protein folding, degradation, and etc., which alters the resistance evolution.
Okay, so now getting to the crux of this experiment. So what was done is a feedback controlled automated platform, a robotic platform was developed where hundreds of E. coli mutant, K12 mutant populations, they were all evolved in parallel. And this robotic platform would control the population size, the concentration of antibiotic applied, and the volume of medium in the culture. They were all controlled. So what was done is this was carried out for 2 weeks, and every 3 to 5 weeks, this robotic system would dilute these culture cells and then transfer them to new 96 well plates. So in this entire process, what was done is, as I've already said, that the concentration of drug, the volume of the media, all the bacterial cultures, they were individually tuned. And it was a feedback control mechanism. That means if in any of the bacterial cultures we observed that there was a decline in growth, then automatically the antibiotic concentration would be reduced so that a minimum 50% growth rate was always maintained. Now, this is basically a graphical representation of what I've already mentioned. You can see that all the cells, they were grown in incubators, vigorously shaken. A strong, selection pressure was provided so that a fast evolution of resistance was always maintained. Okay, now to be able to understand perturbation basically means genetic alterations or deletions. Now, to be able to understand this mechanism, we need to know how the bacteria actually combats antibiotics. Okay, so for this, the bacteria basically makes use of a particular membrane transporter, or also known as a multi-drug efflux pump. Okay, so the one which has been followed in this study is called ACRAB Tolsi pump. Now, this is a tripartite multi-drug efflux pump. That means it is composed of 3 components, and it is consisting of proteins which are encoded by the ACRAB operon. And this operon is present in the bacterial chromosome. Now, so what happens is when this bacteria or the bacterial cultures, they sense a condition of stress in their environment, which is the antibiotic, this operon gets regulated. Now, when this operon gets regulated, the Tolsi, which is the outer membrane efflux channel, the ACRA, which is the periplasmic fusion protein, and ACRB is a inner membrane transporter protein. So what happens, this is a picture of the operon. Now you can see that there's something called a ACR site. Now ACR is a repressor of this operon. So, and there's also MRA binding site. Now what happens is when this bacteria is sensing a toxic compound, which is an antibiotic in its environment, then MRA, which is a positive regulator of this ARB operon, it will dissociate ACR, which is the repressor of this operon, from that site, and it will bind to the operon which will lead to transcription of this operon, and thus the necessary proteins will be produced and the antibiotic which will be excluded from the efflux pump. This is picture given. So what we observed in this study is that if we bring about deletions in these particular genes, we will notice that the resistance mechanism is not being able to be activated in the bacteria. Therefore, there will be a decline in the evolution of antibiotic resistance. So as I've already said, what exactly are we perturbating? We are deleting genes of this efflux pump. We are deleting the MAR-A and TOL-C genes, which will consequently decline the working of the efflux pump, and thereby the resistance evolution will be slowed. Now, lastly, diminishing evolution exhibits, resistance evolution exhibits diminishing returns epistasis. Now, what exactly is diminishing returns? So it's basically a law which means that after a certain point of time in an experimental setup, we observe there is a decline in the rate of output or result, here say, there's a decline in the rate of output, even though we've kept all the factors of production constant. So to explain this in this context, we have to see that I have already mentioned that we are keeping all the factors of production in all the 100 cultures constant. We're giving same amount of antibiotic concentration, we are giving same amount of period of growth, all of that is kept constant. But slowly we observe that there's a decline in evolution of antibiotic resistance. That means the return or the beneficial mutation which we are getting is slowly getting declined. This is happening because there's an epistasis, a strong epistasis interaction between the genetic deletions which are being done along with the resistance mutation of the bacteria. Moving on to Meenakshi.
So now how Prapti has said that it has influences the efflux pump, but not only it influences the efflux pump, but it perturbates diverse cellular functions which also alternate the resistant evolution. It is seen that in this graph, we can see that it is a resistant versus time graph. The gray line shows the reference strain, which is not perturbated, whereas the colored lines are the perturbated strains. So for TAT-C, which is a protein transporter, for LPCA strain, which is the LPS synthesis protein, and for DNA-K, which is a chaperone protein, when we perturbate them, the resistant evolution slows down. But also we see that for certain gene deletions such as MutL and DEC-R, there is an acceleration of resistance evolution. So this also provides us that this experiment is just not limited to the beneficial evolution of the resistance. Next slide, please. So now how perturbating chaperones enhances the phenotypic effect of spontaneous mutation. We know that TOL-C is a client of DNA-K. So when DNA-K is deleted, it knocks out TOL-C. And as Prapti said, it will slow down the resistant evolution. Also, it is seen that for perturbating del LPCA strain, it slows down resistant evolution by interfering with proper functioning of the efflux pump. Now to make next slide. Now to make the study more general, we have used two types of antibiotics. One is tetracycline and the other is chloramphenicol. Now these both antibiotics, though targets the ribosome, it has a different interaction. Next slide. For this study, we have seen that both the perturbating effects had similar and common effects for both the antibiotics. For example, it slowed down the resistance for TOL-C, but it increased resistance for the mutations such as MUT and MUT-L. Thus, the evolution towards an antibiotic is not specific, and hence further studies can be done to investigate more upon it. Next slide, please. So for the basic hypothesis for this experiment was that there is an epistatic interaction between the gene that we deleted and the spontaneous mutation which is happening genetically. So to study this, we have combined our whole genome sequencing and made them into a simple linear regression model, which gave us the estimation of the benefit of each spontaneous resistant mutation. Now we identified the deletion strains and indicated magnitude epistasis, which gave us the quantitative change in fitness in presence of difference mutation. Next slide. To quantify toll C, we have isolated clones from del lac A strain. We have the reference strains, the evolved strains which are evolved in tetracycline, and the deleted genes. Then we measured its IC50 value, and it was seen that deleting of toll C made the evolved reference strain even more sensitive to tetracycline than the ancestral reference strain. Next slide. So together, we can, with this result and with this study, we can highlight that this marks as a potential for exploiting more epistatic interactions to curb spontaneous resistance evolution. Thank you so much.
Okay, students, very nice presentation. Hopefully all the studies will come into effect and we will be able to cure this resistant bacteria, antibiotic resistant bacteria, right? I don't have any more questions. I don't have any questions for you. It was a nice presentation. Thank you.
Thank you, ma'am.
Thank you.
Okay, can I ask one thing from Meenakshi?
Yes, ma'am.
Is this restricted to gram-negative only?
No, ma'am. In this study, it was not shown whether it's gram-negative or positive. So we don't know much about it.
Okay, so they have not specified it for any other, I mean, gram-positive or negative. They are not talking about that?
No, ma'am.
Excuse me. Ma'am, actually the pump, the efflux pump, it's seen in E. coli, which is gram-negative. So they are specifically talking about gram-negative only.
So that is what I think so, because when you were presenting, I'd seen that they were talking about the toll C in the LPS.
Yes, ma'am, but there are many mutations also observed, like the chaperone proteins and etc. So this can be, taken to gram positive as well if further studies are done.
Okay, well presented,
Thank you.
Meenakshi and. Okay, no more questions. I have no more questions to ask.
Ma'am, can I stop sharing then?
Yes, you can.
Thank you, ma'am.
Thank you, ma'am.
মধুমিতা ম্যাম,
বলুন অরূপদা, বলুন।
হ্যাঁ, তুমি ভালো আছো তো?
হ্যাঁ, ভালো আছি।
আচ্ছা, এবার ব্যাপার হচ্ছে তোমরা একটু আলোচনা করে আমাকে ফার্স্ট সেকেন্ড থার্ডটা কোলেক্ট করে একটু আমায় হোয়াটসঅ্যাপ করে দিও। জাস্ট নামগুলো লিখে দিলেই হবে, যে যারা যারা
মার্কস ডিস্ট্রিবিউশনটা কি পাঠাবো?
হ্যাঁ হ্যাঁ,
মার্কস ডিস্ট্রিবিউশনটা কি পাঠাবো না ফার্স্ট?
মার্কশিটটা শেয়ার করতে পারো, বাট প্লিজ কোলেট ইট এন্ড গিভ মি দ্য ফাইনাল পজিশন হোয়াটসঅ্যাপ।
Okay. Arupda, I'll do it. We'll do it together.
Thank you very much. Thank you very much. And for all the students, postgraduate students, you presented well and innovative topics were there. And I also saw the confidence in you because you are speaking from home. So the stage fright was not there. And one thing I really also want to share, that some of the slides you wrote too much, and scientific presentation, it should be more with illustration and less graphics. So don't make it childish, basically. Don't write too much or don't make it childish. Scientific presentation means scientific presentation. So please follow this in future when you are presenting anywhere else. So congratulations to all of you. Over to MC.
Thank you so much, sir. Thanks to all my fellow participants who did so well. Now, this brings us to the end of the technical sessions of today. The feedback form has been provided in the chat box, and you need to fill up the form on all 3 days in order to receive the certificate. We would now like to present to you a short film on MTIM and our cultural event. The film has been edited by Meghna Talukdar of MSc second year and Spondon Mojumdar of BSc second year. In the cultural sessions that will be shown to you shortly, we have Part 1 Inauguration by Shobhik Bhattacharya, Pushpam De, Tanisha Chakraborty, Stuti Barua and Rohan Dan of BSc Department, and Meghna Talukdar, Elizabeth Basumatari Arena, Devdatta Das of MSc Department of Microbiology. Following that, we have a dance performance by BSc second year featuring Shinjini Datta, Niti Chaudhuri, Sanjana Lahiri, Akanksha Shaw and Shirsha Mukherjee. And at last, we have a dance performance by BSc third year featuring Sneha Das, Rajeshwari Chaudhuri Nath and Shreyoshi Bhattacharya. Looking forward for the cultural vibrant event. The link for the cultural event will be provided in your WhatsApp groups sharp at 9 p.m. See you all shortly in the cultural show. Thank you so much.